experimental. SciAnts. We're curious, non-judgmental. Always good vibes when science on screen. Time to learn live with SciAnts streams. SciAnts. We get experimental. SciAnts. Curious, not judgmental. Always good vibes when science on stream. Time to learn live with SciAnts streams. Welcome to the SciAnts family. Happy to meet ya. Let's introduce our scientists. There's Bullint and there's Lita, mom and dad up in the lab. The dream team in their heart, they share a passion for wholesomeness, for science, and for art. Science is for everyone, all backgrounds, all ages, all genders, and all walks of life deserve an education. Come be one of us. Spark your inspiration. Art and science are one. They're a conversation. 7 days a week. The fun is interactive. Science experiments done on stream are so fantastic. You'll see microscopy. It's super dope. It means checking out tiny worlds under a microscope. You'll learn about ecology, neurobiology, evolution, and of course bug out on entomology. Get the latest scoop of science news when you stop by. We also break down sci-fi movie science on the live. Antonia, the leafcutter ad, or Adacephalotes, stars in our book game and she's the Twitch emoji. You'll meet cannoli, noodle, hops, and barley too. When there's a hype train, the cats can party with our crew. Inherently important, and what we truly cherish is community support. Wholesome expression ferret keeps it wholesome AF with kindness in the chat, because our crew is the best. That is true, that is fact. SciAnts. We get experimental. SciAnts. We're curious, non-judgmental. Always good vibes when science on screen. Time to learn live with SciAnts streams. SciAnts. We get experimental. SciAnts. Curious, not judgmental. Always good vibes when science on screen. Time to learn live with SciAnts streams. Yo chat, who's ready to learn with SciAnts streams?
Oh, hi everyone, and welcome on in. Happy Friday. Y'all, it's that much closer to Monday. Spikes! Spikes, thank you for the resub. Tier 2 for 19 months. Thank you, Spikes. On a 19-month streak. Sir, you're approaching 2 years rather rapidly. Thank you, good sir. Hi, everybody. Welcome on in. First today was Queen Yoga Garrel. Queen Yoga Garrel was, in fact, first today. Second was Cliff. Hit it! Music Girl, thank you for the 100 bitties. Music Girl, thank you. Jimmy, there's so many chaos causers here. Jimmy, thank you for the 100 bitties. There is so much chaos here. Y'all are being so naughty. Speaking of naughty, I did not get my hair cut with 34,000 channel points on the line. I did not get my hair cutted with 34,000 channel points on the line. Chat! Before I continue welcoming everyone in, I'm going to set up the next prediction of what is the new species of the week. As we always do, let me just set up that prediction, then I'm going to welcome everyone on in. I just want to have that prediction going while we're doing the welcome. Let's see. New species of the week. All right. Hi, Ubi. I'm just writing this out for the new species. Louie, indeed, Ubi. Let's see. What else was it? All right. There we go. Alright, y'all have 5 minutes to get those predictions in of what's the new species of the week that we're talking about today. Get those predictions in. Ubi says, or Smike says, you didn't do the level 1. There is a raffle spiel. Oh, thank you, Smikes. Appreciate you. Guys, we're on a level 1 hype train. If we complete a level 1 hype train, then we will do the next thing, which will be to do a raffle, chat. Raffles for stickers and or pins and or magnets. All the things are possibilities. Thank you, Master Smikes. I should bring it up on the screen. There you go. There is all these fancy stickers, magnets, and more that are available off the menu if we complete any level of the hype train. If we complete level 5, we'll do pins, and I'll put on a ferret onesie. But never any pressure to get us there. Now, let me welcome everyone in. Of course, there was Yogurt and the lovely Cliffhouse McLean. We got Restock and Skip. Skip the Pickle, a.k.a. Pickle Skip. Welcome in. Darth Locutus, our first-time chatter from our plain old Trey crew. Darth Locutus, welcome on in. I hope you're doing well tonight. Again, Ubi is here. Hello, Ubi. Scrolling, making sure. Peter, welcome in. Toggle, hello, good to see you. Optic Nerve, Quoth the Raven Nevermore, being safe, I hope. Quoth the Raven. Noki, hello, good to see you, Noki. Luminology, hello. We got Music Girl and Jimmy with the biddies. Thank y'all very much. Jimmy, I heard I maybe made you chuckle. Maybe. Possibly. Did I make you chuckle, Jimmy? Did I make you chuckle? It was recorded in Portland. I'm sorry, Ubi. Guys, also go follow our friend Jimmy B93, who I may have gotten in trouble today. I may have sent him a TikTok that was not supposed to be sent to him. And he may have been ever so slightly chuckling about it. To which he may have had to show Yogurt Girl, or Music Girl. Maybe Yogurt Girl as well. And Music Girl was not impressed. Music Girl was not impressed. The hilarities that were ensuing at those breathtaking tiki-takas that I sent. Supreme Doom, welcome in. To the amazing artist that is Supreme Doom. And Shipwreck, welcome on in. How are y'all doing today? Supreme Doom, I had no idea it wasn't spelled correctly. I just assumed it was spelled correctly. I'm like, that looks good to me. I recognize the bonjour right there. Doing great. Excellent, guys. We're going to get a fancy shout-out for Supreme Doom in a moment as well. And for Cap Coder as well, who's going to be satiated today. Oh, my goodness gracious. My God, Captain Coder. Hello. I hope y'all are doing well. We are in the midst of a level 1 hype train. Remember, if we complete the chew, any level, we will do some raffles for some stickers, if you so choose, but no pressure at all. There's also a prediction right now of what the new species of the week is that we're going to kick off the stream with. It is Supreme Doom! Thank you for the 300 biddies! Thank you, Supreme Doom, for the biddies. Supreme Doom. The usual kickoff we'll be doing. Captain Coder! And Nate Kind, thank you for that follow. Welcome in. Captain Coder, thank you for the 1,000 biddies, Captain Coder. And Toggle, thank you for 200 biddies as well. My God. Y'all are kicking off. Y'all wanted the chew. Y'all got the chew. We have to do some raffles now. There are new items. You may not have seen these items before. A couple of new magnets. Shipwreck, thank you for the 301 biddies. Thank you so very much. Let me go ahead and set up the raffle. Alright, it is now live. Nate Kind! Welcome in. I have set up the raffle. Tackle, thank you for the lurk. The raffle is now live. Exclamation point raffle to enter. If you win, I will need your mailing address of where to ship off to, but it's exclamation point raffle to get up on in there. And then we will, I'll send it off to you.
Together, we can rule the galaxy.
A truck horn, thank you for the resub. That means we're going to get 2 raffles tonight, at least. A truck horn, thank you for that, converting that prime sub to a tier 1 sub. Appreciate the heck out of you, truck horn. Dandy him, hello, how are you doing today? Welcome on in. As well to you, a truck horn, I hope all is going well. His exclamation point raffle. To enter in the raffle, in the meantime, make sure to do a couple of things. First of all, follow our lovely friend, Supreme Doom. Amazing artist extraordinaire. Just like the coolest handmade cards and speedrun cards. It's mind-boggling. Hello, Michael R. Hassler. How you doing, Michael? Welcome on in. Good to see you, Michael. The prediction has now closed. The prediction is closed. I will fulfill the prediction once we've gotten and we finish the hype train.
Together, we can rule the galaxy.
Nate Kind! Nate, you just came first time! You follow, first time chatting, you subscribe. Thank you so very much, Nate Kind. Appreciate the heck out of you. For anyone who subscribes, remember, you get access to sounds and special gifts that you can trigger. So you have access to all of these sounds, these gifts, and then you also have access to gifts too. Electric Boogaloo. So make sure you can you have fun and play with those. Thank you all very much for that subscription there. And no, Supreme Doom, thank you, Supreme Doom, for being part of our community.
Gucci.
Gucci indeed. Gucci indeed. So I hope everyone had a great day today. I know I was very busy. Baby Ilona and I spent the whole day together. I've got her baby monitor here. So if we have to do a BRB, it's because I gotta go check on her. Lita is out of town tonight. She'll be back later, like late tomorrow night. So it's just me and the little one. So we were busy working on a way, and then we all like she was here with me during the day as we were working. We went on a walk. We played a lot, like all kinds of sillies she was having today. She really likes rolling around on her floor. And yes, she did see Master Smikes today on FaceTime, and she was smiling ear to ear. It was just this, Smikes was chatting with me, but the camera would tilt, and then he would just smile at her, and we're like, "What are we talking about again?" And it's like, yeah, that's right. Like, she's such a cutie that you just forget what even you're talking about. You did, and I think it worked. Although she seems to really smile at anyone who has a beard, and then. But she was like super into Smikes as well. So Jimmy, you've got competition. That's all I gotta tell you. Jimmy B93, you might have a little bit of a competition. Speaking of competition, folks, the Captain Coder's in his house. The Captain Coder, educator extraordinaire. He teaches people about programming, game development, really whatever comes up to mind in a very accessible way. Not in a like demeaning at all way, but like.
My name is Gwen Stacy.
Hi, Gwen.
I was bitten by a radioactive spider.
Very accessible. Insatiable.
Sisters of fate could not hold. And you will not see the end of this day. I will have my revenge.
Hi, nerds. Hi, Gwen. Hi, Gwen. Guys, go check out Captain Coder. He is insatiable. He told me I was insatiable. Perhaps he needs to be satiated tonight as well. I love your face, my friend. Seriously, y'all. Amazing educator. I've never heard this. He refuses to go to be affiliated because then something might be behind a paywall. So he is unaffiliated. He would be on a partner push, full disclosure, if he became affiliated, because he's that well attended and people are excited to be in his stream, asking questions about coding. One day, Captain Coder, we're gonna turn you to the dark side, and you're gonna have that purple check mark next to that name. I tell you this right now, it'll be the fastest and like partner push we'll have ever. Maybe that's why he's so insatiable. He just wants to become affiliate and partner in the same week. If anyone can do it, it's Captain Coder. I have a sneaking suspicion now. Guys, exclamation point raffle again to enter. We have 20 seconds left on the hype train. Any level will complete, we will do raffles. We will do at least two raffles for sticker and or magnet if you win. Remember, I will need a mailing address if you win so that I can ship this out to you. And if you haven't won in a while, then I will ask you to resend your mailing address because I may or may no longer have it. And thank y'all again for that hype train. Far too kind, y'all. Far too kind. Kicking off the stream. We hadn't even done anything and we had a hype train. Thank y'all very much for that. Alright, let's go ahead and pull our two raffle winners for today. Is there a sticker of the magnet? The word magnet? No, unfortunately, Ubi. Fortunately, there's not yet a sticker of that. But the Twitch words one, there's a stickers of. But then these are only magnets currently. Yeah. Alright, in 3, in 2, in 1 for the first raffle is... Shipwreck! You broke a toe! Dan, I don't like that. Dan, did you get it patched up? Shipwreck! Shipwreck, come on down to the front of the line. Shipwreck V27. Shipwreck, congratulations on your sticker or magnet. Which one would you like? 1 through 13 are stickers. 14, 15, 16, 17 are magnets. Shipwreck, what would you like? What would you like, Shipwreck? Remember, y'all, we do ship international, so you do not have to worry about any shipping charge. we send it straight to you, and you're all kinds of Gucci. And you shouldn't have to pay any import tax either. The only time you have to pay import tax is if we ship something outside of an envelope, like a pin, and that's only some countries. 14, partner magnet. You got it. Shipwreck. Would you mind please DMing me your mailing address either on Twitch, Whisper, or Discord DM, and we'll get this shipped out to you. All right, y'all. Number two, two, in 3, in 2, in 1. Luminology! Luminology coming up to the front of the line. Luminology. Luminology, congratulations on your winning. What would you like today, Luminology? 1 through 13 are the stickers. Sticker number 5 is the wheel spin, and again, 14 through 17 are the magnets. What are you feeling today, Luminology? Do you have no? I have no idea. I'm so sorry, Luminology. If I had that level of memory, I don't know what I'd be doing. Oh, no worries. But I guess I do have a record of all the winnings handwritten down, so we could, in theory, go through and find which ones you have. Although the numbering system has changed as well. Kevin Drums, you got it. Alrighty, thank you all very much. Appreciate the heck out of you guys for that hype train. Let's go ahead and get into the new species of the week to kick off the stream. I will this prediction results, and let me bring up the image to get the right paper. Okay. Let's open up the figure from the paper. Ah, perfect. Good deal. All right, so the new species of the week with 6,155 channel points on the line. The correct answer is a leaf bug. This week, that has been the new species as a leaf bug. 6,000 channel points go to, it's Jimmy B, 93, Jimmy Bet, 420 channel points. 420 channel points, Jimmy B Bet, on a leaf bug. And Jimmy won. In fact, this is a loaded term because it's not just one that was discovered. Yes, so this is actually a true leaf bug, Smikes. We're going to go into it. Capitalism Vice, love your face, Jimmy. Isn't much that can be done. It's not the big toe. I do have a podiatrist appointment on Wednesday. Take care of yourself, Dan. Please take care of yourself. All right, so y'all, in fact, it's not just one leaf bug that they discovered. They actually discovered 7. They discovered Pookie Bear. Welcome the heck in, Raiders.
SciAnts stream, but we ain't bugging. We over here bubbling. You be chugging what we got. Now the tribes arrive. I promise that I have prescribed you vibes. The vibes we got are giant. Shout out to the stream for the raid. That's SciAnts.
Well, Raiders, welcome on in. Pookie Bear, and Shipwreck, hello. Pookie Bear, what the heck were you doing today in today's stream? I see there were some makers and crafting. What were you making? What were you crafting? I want to know all the things. I have, let's see. Oh, I have the Instagram. Yes, I have your Instagram up, Pookie Bear. I have your Instagram up, ready to show off. Get followed on, oh my god. You do amazing things. Good, diamond pay, oh my god. And I see the miniatures that you make. Oh my god, Pookie. You're in the right place. I'm an ant geneticist, and this is I'm geeking out, Pookie, at how cool your Instagram looks. Guys, I'm gonna give Pookie the big fancy shout out when I start showing y'all the pieces they were working on. But Pookie, if there's anything that you want to share from today's stream, feel free to drop a link into the chat, or you can drop an image or what have you into the Discord. Anything is Gucci, but I do have your Instagram to show off, and anything else that you would like to have shown off. For the Raiders coming on in with Pookie Bear. My name is Blunt, 1/3 of SciAnts streams. The other 2/3 are my lovely wife Lita and baby Ilona. We're research scientists and we do interactive science streams here on Twitch most days of the week. Microscopy art, sci-fi science, science news of the week, deep dive scientific topics, games, 3D printing, IRL streams, and more. Raiders, welcome on in. We're just kicking off our science news of the week. But before we go anywhere, this Pookie Bear's Instagram. Y'all, this is gonna blow you away, this artwork. These are so freaking cool. There's the fancy shout out as well. Guys, check these out. The miniature painting and miniature, like, bird's nest. These are so cute and so well done. Pookie, I see people do these, like our friend Corn Relish has done these as well. And I don't know how people do it without a microscope. Yes, Ubi, this is furniture for ants. That's why I got excited. That's why I got so excited. Look at how good they look, Ubi. They look really, really nice. Like, this is, like, nicer furniture than I have. Check out this, the baker shop. Look at all these. And they're filled up with there's even little bread in there. It's an entire, I know, Supreme Doom, listen, this is like a, this is like a posh ant right here. Most ants just live in the dirt. This one, it's got a bit of everything. Miniature flower shop. I did a miniature bakery shop too. This one is so cool, Pookie. I love the little bread pieces in there, and just like the extra details that are going on. This is really, really cool. Yeah, like look at all this detail. Like even the flowers, the papers, the stapler. Even like the little, like the pizza dish here. Pookie! Thank you for the follow, Pookie. Welcome on into the colony. These are absolutely amazing, Pookie. What? Oh, and there's, yeah, look at how tiny they are. It's so cute. These are so cool. Shipwreck, I get it. It is super tiny, but I really, really like these. These are so adorable. Well done, beautiful. And you said you were diamond painting tonight, Pookie. What were you diamond painting? We have a diamond paint piece in Little One's room. Our friend Coffee Rocks did 2 diamond painting pieces for baby Ilona of Winnie the Pooh. And those, they're really, really amazing because if you like look really close, they just look like dots, but then you back away and you lose the, like, any semblance of dots, and it all kinds of blends together. And it's just really remarkable of what you can do with diamond painting. So Ubi, think of it like tiny colored dots. And when you back away, you get a full image and you don't really see those dots. It's really, really cool. What is this? Instead of ants, how can you expect it to teach children to learn to read? You can't even fit inside the building. A truck horn. Diamond painting a cat. Ooh, Pookie. Was it your cat, by the way? Because I know you can get custom order diamond paintings. Yes, if you add it to Insta, please let us know, and I'll happily show it off. Or you can throw it on the Discord as well, whatever is easiest for you. Thank you again for the raid. Before we kick off, I forgot to start a new prediction. For the Raiders coming in, today is our science news of the week, and one of the predictions that we always have is how many topics will we finish tonight? And I'm notorious for going slow. Chat's notorious sometimes for voting for more. And we'll eventually figure it out. We'll eventually find a balance. You can vote on how many topics we'll finish tonight. Hi, Emma. Jimmy, Jimmy, I don't understand your redemption.
I have a good mind to go home. You had a good mind, you wouldn't be here in the first place.
That's fair. Jimmy, so y'all, a little backstory. Satan Bird is a character on Jimmy B's stream. He wants a... So does that mean he wants a praying Antonia? Is that what it is? Pookie, excellent. I will refresh in just a moment. We just had 100,000 channel point redemption, and I want to make sure that I put in the right thing, and we get Jimmy the correct redemption that he wants me to do. He wants a penguin Antonia. Like an Antonia wearing a penguin suit? I know Satan Bird as a... Ubi, Satan Bird is a penguin, and I think Jimmy wants... Okay. Okay. This is gonna take some imagination. Jimmy, thank you for that 100,000 channel point redemption. And thoroughly confusing me as to how I will make a penguin Antonia. But we'll figure it out. Guys, this is the diamond painting from Pookie Bear. This looks lovely. So, Ubi, you see how there's these little circles? When you back away from it, you don't see the dots anymore. It's really remarkable. The Pingu shocked face at the end, but we'll figure it out later. Hold on now, Jimmy. Jimmy, hold on. Music Girl said you wanted it as a penguin. Now I'm thinking of it if Jimmy wants it as the penguin from Batman. Yeah, Peter. Yeah, he wants the Newt Newt. He wants the Newt Newt. For those of y'all who don't know, I will quickly derail, and I will show y'all what we're chatting about. Hold on. Not the eDNA, leaf bug. Okay, Newt Newt. This is what he wants. Yeah, but Jimmy, Music Girl didn't say if it says you want that face, but do you want it as a penguin? Guys, this is the Newt Newt Jimmy wants. I mean, this is hilarious to me. This is a redemption on Jimmy's stream. And I chuckle every time because he's always like, praise Newt Newt! And it just makes me smile. Every single time. Last face was as Antonia, and you don't want it as a penguin. Oh, wow. Okay. Okay. I think we'll try it. Did you know there's this one too, Jimmy? What? Dark Souls theme. All right, we're gonna figure this out, Jimmy. Antonia penguin suit? Yeah. I mean, well, Music Girl, we put Antonia in the Fall Guy suit. I imagine it's not going to be too different from that. Right? So that if that style is okay with Jimmy, we can do that. We can absolutely do that. Now, Jimmy, we have to put this music to it, though. If we make it an animation, we have to throw that music on there. Yeah, exactly. Ubi, like this one here. Okay, we can do that. Yeah. I feel like it's going to be madness, but we can make it happen. And then we have to have the Requiem music playing over it. But, Jimmy, I draw the line that the command will not be !Satan. It is not that kind of stream. It can be S-Bird as a deep cut reference to you and my sillies on your stream. Or yeah, it could be new. Yes, but I will need you to remind me of that, Music Girl. Now that you've said it, I'll probably have a more likely to remember. Two butts is different, Smikes. Don't you dare use the word name two butts in vain. What? Didn't make any sense right there. Chat. Thank you, Music Girl. Pookie, you have no idea what you just rated. I appreciate the heck out of you for coming on in here. We are talking about the new species of the week to kick off the stream. I did, Music Girl. I do love the Two Butts. Guys, if you don't know, the creator of that Two Butts song is our friend Giraffe Killer. Giraffe Killer gets his name from he was in Africa protecting giraffes, and from poachers, that's hence his name. Please go drop him a follow. He's an absolute legend, a really kind guy, and he is, if you do see him, please tell him that he is kind, wholesome, and handsome, because if you tell him that on his stream, he will jump out of his chair and run away. And it's absolutely beautiful. Absolutely beautiful. Thank you. Yeah, devastating disease of truck. We're devastating. Speaking of devastating a truckhorn, we were almost having devastating news about these new species. Look at that. I took us right back to the point of the stream. I'm getting better. So, hold on. Hi, Nelf. Welcome in. Hi, Nelf. The fish show can't. You got it. You got it, Optic. Nelf, we just had 100,000 channel point redemption for a custom Antonia, and now Optic Nerve just dropped a deep dive request for fish hook ants, which are terrifying. Nelf. There we go, and it is in the sheet. Thank you, Optic, for that redemption. A leaf cutter ant's fooled by the leaf beetle's camo, or even threatened by him. So, luminology. So this is a leaf bug. It is in fact not a beetle. It is actually the nomenclature is quite interesting. It is what is called a true leaf bug. So there are leaf bugs that are more closely related to stick bugs. So those are called. That is the phasmid group. That's how they're subdivided. And then there are true leaf bugs that are functionally different than what you classically have seen. And so the difference between the true leaf bug and the leaf bug that is more like a stick insect is a lot of the body morphology. So these, because they're the true leaf bug, they actually, the way you can tell is that their abdomen is much more flattened out and it's much more width of a leaf, versus the ones that are more closely related to a stick bug are more kind of pressed in together, if that makes sense. So it's your morphology is a little bit different. Mind you, there's also genetic differences as well. So when you're doing the sequencing, they get categorized into different groups. Right now, with stick and leaf insects in general, there's about 3,000 species. And then there is the true leaf insect, or leaf bug, that we're chatting about today, and that's a different, a subgroup of these phasmids, and they're called the Phyllidae. Phylliidae. And these are primarily Asia, Australia, and in the Pacific. Until this paper came out, which this paper came out, y'all, let me make sure I get it right, the 3rd of August. So a very recent paper. Not Philadelphia, no. And luminology, the mango martini sounds delicious. Before it came out, this paper came out, it was thought that there were very few of these species. In fact, it was thought that there was one species in India of these animals, and it was fine that their populations were decreasing in different parts of the country because there are so many of them. Well, a group of entomologists went out and did a bunch of field collections in Southeast Asia. And we'll talk about how they did the collections, but you can see here there's visual differences between these animals. Again, they have the true leaf insectness of them is because they're flat body, and they also have the leaf vein patterns that you would see. And what's interesting too, and so these are actually the wings here, and these are female specific. Leaf bugs are not true bugs. True bugs are under Eumetobola, while leaf bugs are under the sister group. Yes, Cliff. But so I guess there was a nomenclature difference then, Cliff, because there was, or maybe it's something lost in translation, because the reference that they were using was the true leaf bug versus the ones that are more closely related to the stick bug. But you're right, there's another sub-difference there of the true definition of bug is also separated out from it. So maybe the better way of saying it would be leaf insect. So true leaf insect versus leaf insect, I suppose, right? I think that's in the paper they were using bug, and there was an interview as well, and they kept using bug. But you're absolutely right, Cliff. Absolutely right. But I just again wanted to highlight y'all these. I thought it was really cool that each one of these samples here are female, and their big delineator was that here are the wings of these animals, and the way that they're these true leaf insect is that they have these leaf veins. So you see how well their mimicry works with leaves is because, again, they have those leaf vein components on each one of these insects. Even though, you know, they're functionally looking pretty different in terms of color and maybe some texture, but at least the leaf veins are similar, and again, it's that really flattened out abdomen. Another way of telling that it's female is that that abdomen is quite on the wide side. And we've seen in the past with insects of all kinds, the bigger abdomens are akin to female. How big? Ubi, what's cool is, so this is a Zukeys paper, so we're going to see the measurements for each of these. So there's going to be, I'll show y'all how the table looks, because we're going to see some common themes of how these papers are done. And you'll see, Ubi, there's good papers of these, and then there's like the standard. And this is more along the lines of the standard papers, meaning that they do some morphology or structure analysis, and then they'll do their basic sequencing to show that genetically there's a difference. And then that's kind of that. Versus we've had some new species papers that have actually looked at, hey, we found this genetic component, let's see if it's actually responsible, and then they'll do a mutation for it. Should you be afraid of these? No, Ubi. So these, there's no evidence of toxicity in these animals. Really, like, and you know, because and hi, Wind, saying, welcome in, Wind. How are you doing, Wind? Your big thing would be, Ubi, right, is if they had like really bright colors, if they were trying to show off some level of poison. But because they're trying to blend in, that is already a good enough hypothesis to start with, is that they probably aren't trying to be seen. So they can't flash and say, hey, I'm toxic, don't get near me. They're trying to hide. And why are they trying to hide? From predators, Smikes. Don't worry, they're not hiding anything as far as I'm aware. So, what they thought, the main predator for them, birds, and then there's other like bigger insects as well, and also small mammals will also go and eat them. No, so a truckhorn, they are being population-wise devastated, because even the entomologist who led this study was saying that by eye, individuals of different species usually are counted as the same species because of appearance. So appearance alone tends not to be different enough to differentiate them between different species. And so in India, up until this paper, it was thought there's maybe one or two of these species in the country. And because some populations were decreasing across the country and others weren't, you know, they were like, well, it's fine because there's two species and there's still bunches of them, so we're good. And that had been kind of the mindset of that Pacific Southeast, from India, Philippines, and even to Australia. Yes, the bugs are saying, don't get near me. Don't even see me. You haven't seen anything, Ubi. But truckhorn, they found 7 new species. And again, there were and 4 of these, 4 of the 7 were from India. 4 of the 7 were from India, and they were different species that had never been identified before. And they had actually been grouped into one species before this analysis. And so they found completely new species of leaf insect. And this, what they were saying was, so the authors had this quote, not from the paper, this is, they did a press release. And this is what the lead author says. The finding is important for species conservation. If all individuals die out in India, it is not just a group within a species that it was reduced, as previously thought. In fact, a whole distinct species is being wiped out. This means that the Indian species is particularly important to protect, and species there being plural. They also go on to say there's 3,500 known species of stick and leaf insect in the world, but there are only, of those, 100 of these true leaf insects described. And so it may be a gross underrepresentation, because they were saying a lot of this is very difficult to do in classic morphological studies. So Ubi, you asked like about sizing. So if we go to the actual paper, in the paper they do your standard measurements. First they do the imaging, which actually I thought the images were really, really pretty. I thought they did a beautiful job getting nice images of this, especially like the delineation between of the wings. Like honestly, if you have a low res image, I don't think you can tell that's a wing, and seeing the leaf wing veins, but they oriented them all the same direction and they lined them up on, it's a really pretty figure. And I can see like physical differences between the animals. They then here Ubi have like of how they collected the samples, which we've actually criticized in the past of different papers and how they did it, because if you're sequencing, if you're trying to prove that you have a different species, it's important to know how many samples you're sequencing. If you're only going to sequence 1 or 2 samples, right, then are you actually finding something different? Or did you just find a different species, or are you just finding an individual that has like a mutation or 2, right? So like how many samples they validate on, and then like the really good papers that go above and beyond do like male and female samples as well. And they'll also do like mitochondrial sequencing to compare mitochondrial genome as well. For these, it was quite interesting. Apparently they tried to find features per each species that demonstrated whether or not they're male or female. And apparently, if you have a species, some species are very easy to tell male and female apart, but because the different species look very similar to one another, in other species it's not easy to tell them apart using the same technique, and so it gets very murky. And so really, the best way, it turns out, to identify without a doubt these species is to dissect them and to then find out if they're male or female. So it's quite interesting that it's so difficult to tell. And again, it gets back to the argument of, because they're so similar in appearance, even if you have one rule that works for a given species, because you'll be looking at them and living and cohabitating in the same population, you may not be able to sex other individuals that again look super similar to them. So they did make the measurements here. Ubi said they made it to the nearest millimeter, which I think they did quite a nice job on. They even like go into talking about like what kind of clampers were used, what kind of cameras, what kind of lens. Like they really went above and beyond in their materials, more than we've seen in the past. The photography, the laboratory work, and what kind of analysis they did. You need a universal translator to ask them. Maelstrom, you're not wrong. You mean to ask the insects the sex? Yeah. And it's because if you have to dissect them, right? Like that's definitely a tell-all way to do it, but then you can't set up mating experiments. You can't set up behavioral experiments based on sex, right? Because you had to rip up their ovaries or their testes. And there's no coming back for that, at least not in current technology. So it's, if you had a translator to ask them male, that'd be the way to do it. Here's the phylogenetic tree. We won't go too deep into this, but it's essentially a family tree of relatedness, and they've marked in the highlighted green the new species that they've identified. And what I think is kind of nice is that these new species are not all clustered together in one particular zone of the phylogenetic tree, but rather they're spread out across the tree. And why that's kind of cool is it's a sanity check to make sure that, hey, in fact, we are hitting up different species. If they were all clustered together at the same point of the phylogenetic tree, one could say, well, maybe you did a poor job in your sequencing and you're just like over-sequencing something. Spectre, how dare. You could be over-sequencing something and that's leading to these differences. Spectre Flight, can we wait for the end of looking through the paper? If you run away, I'm getting rid of the redemption forever. Spectre Flight. If you want the redemption to go away forever, then that's what you do. I warned V about this. V did this all the time. He would hit it and run away. Poochie lip, that's fine, Spectre Flight. I can deal with the Poochie lip. I can deal with the Poochie lip, Spectre. All right, we'll go back into the paper in just 1 second, folks. We have been interrupted by the lovely and amazing Spectre Flight. He wanted the ferret. Hello, Spectre Flight. Hello, everybody. My name is Wholesome Spectre Flight. I'm here to tell you how beautiful, amazing, merry people. We began with the Redeemer. Spectre Flight. Chad Spectre Flight is the purveyor of chaos. He is also the creator of the Hyosai Ghostbusters mobile. It will be running around in Las Vegas. We're going to TwitchCon. It's very, very cool, very, very hip. It's very, very nice and lovely peoples. And he's got beautiful cosplay of the Mandalorian the people. It's very, very true and very fact. Yes. We have a Smike 24, who's one of my best of friends. He will be one day neighbor to Balint. They will do the Hyosai barbecue item and have the Hyosai the beverage item across the fencing. It'll be very, very beautiful. Smikes, how is my brother Mendel Farah doing? It's very, very true. We have Music Girl 95. We have Music Girl 95 is Hyosai educator of the children. Music Girl is the purveyor of truth and knowledge and all things cool and hip and fancy. We very much love you, Miss Girl. It's very true. And we also love boyfriend, Jimmy B93. Chat Jimmy B93 is Hyosai. Breathtaking, beautiful, handsome gentleman who is the leader of the concert hall. Jimmy is a Fall Guys hero. And he is the kisses of the duck song. It's very true that he does these. Chat, love the Jimmy B93 who will one day come to TwitchCon. And then we will dance on the TwitchCon stage together. And we'll go bam, bam, bam. Got any grapes. It's very, very true and very fact. Yes. Chat. It's all the artworks, chat. If you did not know this, Ubi is Hyosai Ubi. And it's amazing. Does the writing and the children's book illustrating and is extremely handsome is the Ubi. Chat. I will now ask you if anyone else wants to be hit by the ferret. If you do, then I'll come back and return and tell you how the breathtaking, beautiful, and amazing peoples. Otherwise, I wish you to have a great stream. Very true and very fast. Yay! Thank you, Spectre. Spectre Flight. I hope you appreciated Spectre Flight. I hope you appreciate that, Spectre Flight. Morgana Freya, hello. Torrid's Night, hello. Welcome on in. I hope you're all doing well tonight. Morgana, beautiful artwork tonight, man. Absolutely stunning. Guys, a couple of shout-outs. We do have the lovely Morgana Freya in his house. Morgana was doing amazing artwork today. She was doing some clay sculpting of a dragon, and she was nailing the structure of it down just beautifully. And she was like, and listen, y'all, she was being, I would say, misbehaved a little bit because she was like, oh, no, it's nothing. It's fine. It's not a big deal. And no. How dare, Morgana. It was looking beautiful. Again, the outside texture of it was spot on, as it always is. Thank you for sharing your art with us, by the way. So for those coming on in, we're talking about a new species of true leaf insect that was discovered. Actually, 7 species that were discovered this past week. And here is the phylogenetic tree. That is the tree of life. And it's like a family tree that it's looking at for relatedness. And it's these ones highlighted in green highlighter. are the new species. So there are 7 total species that were discovered. I really, really like this image where they show 3 of them. These are called the true leaf insects because of their flattened abdomen. Not beetles, Spectre, not beetles. So they're closely related to stick bugs and other leaf insects. They're all clustered together in the same group. This is a subgroup of those, and the difference that you're seeing really in these animals versus your stick bug and your other leaf bug is the abdomen is much flatter in these animals, and then if you zoom in on the wings, so what I didn't notice until I was reading the paper is if you look closely here, this upper outline are the wings, and the wings have the leaf veins like you would see on a leaf. So it's an amazing piece of camouflage that these animals have. Now, the unfortunate and silly thing that we were talking about earlier tonight, folks, is that these animals, unfortunately, are being hunted to endangerment and or extinction because apparently they're very difficult to tell by eye that they're different species. And so, India, the survey that they did, thought there was 1 species, 1 to 2, and now there's actually 3 to 4. And yeah, it's quite wild that there's so many that they're actually present. The only way they're finding them is through molecular evidence. So doing the genetic sequencing. So we're just going through the paper and what were elements of the paper that were demonstrating these components. Spectre says, caught a wild juvenile mouse, now we're raising it because hard too big. Yes, Spectre Flight, I understand that. That's fair. Where are these found? So Grimly, these are found in the Pacific Southeast. So India, Philippines, Malaysia, those areas are where they surveyed. And they did make a point in the paper to suggest that, it was a pretty decent survey. They took a bunch of samples, they did sequencing, and they identified a bunch more species than what they thought they would find. And they suggested that of the leaf insects that have been discovered so far, if we went back and did additional sequencing, there's a possibility that instead of having 100 of them, there might be 200 or 300. And then on top of that, there's probably other species of leaf insect that we haven't even come across yet. So their population is probably much more than what we give it credit for. Same thing with dispersal too. They were saying that even ones found in other parts of the world, there could be different densities of different species instead of just having like 1 or 2 in a given area, which I think is also quite interesting. Does that make sense, Grimly? Guys, also go check out our friend Spectre Flight. Gamer extraordinaire is the lovely Spectre Flight. Go check out Spectre Flight as well. Chaos lover too. Awesome, thank you, Grimly. So the next additional segment that they did, they got something called a holotype. So we've talked about what a holotype is in the past. It's a singular example of what represents a species, right? And that's really cool because if you identify a new species and then someone else finds a species as well and they want to go back and take a look at your sample and compare it and see, okay, did I really find a new sample? Is it the same sample as someone else found? You have this thing called a holotype. And it's like, here, this is what we're basing our measurements off of, and this is like your number 1 primary sample to go after. There's, of course, other samples that you're also going to be sequencing, but here, say this one, for example, deposited in the Montreal Insectarium in Quebec, Canada. So if you want to go look at the first one that was described, you can go to the Montreal Insectarium, and you can see that there are 9 females, 6 males, 1 nymph, 1 male nymph, 2 female nymphs, and 6 eggs. And that is what they have to be able to look at and see, like, this is that species, which is quite cool. They also then will talk about some structural differences and structural similarities between the animals. So again, it's just getting back to functionality at this stage. So it's showing some differences in terms of body plan between species. I like these views as well because you have to try to find the leaf insect. And full disclosure, y'all, I have a lot of trouble finding them in these images because they do a really good job in blending in. here are their eggs. Oh, the eggs look kind of cool. So from two different species. And yeah, I think it just looks really pretty. It's a unique kind of egg. I didn't realize the eggs looked anything like this. And it's just a really nice, like, zoomed resolution that we're able to see in here. And so there's a lot of texture. We don't have any information, unfortunately, Supreme Doom, as to why like the textures look like this. You know, like there's these little indents around here and then these little hooks around here. We can hypothesize and suggest that these hooks are for like staying onto a particular substrate, but then, you know, these like that are a little bit smoother, that, you know, like I wouldn't know offhand. Yeah, these are egg spectra of different species of these leaf insects. So these are 3 that have not yet encountered water, and then these have encountered humidity. And this is another cool feature of the eggs. They haven't gone into the biology of these a lot. While these are 2 different species, they have in the supplement, which is unfortunately behind the paywall, they have different species as well, of ones that will be exposed to water and not, and they change shape. Versus arboreal. So, unfortunately, Spectre, a lot of the paper doesn't go into really living areas. Like, they talk about where it was collected in terms of physical location, but not so much if, like, you know, all of these are in the trees or all of these are like on the ground. My bet is most of them are trees though, because they're blending in with leaves, so like some level of tree one. They were all eggs. They were all eggs, Spectre, from the leaf insect. And then here is the nymph stage. So here is an early stage of development. This is more aquatic animal. It's not clear if you can sex these at all, but you can start to see the beginnings of the body plan. The pick of the third. What do you mean, Spectre? So these, the nymphs, again, they look quite different from the adult. You don't have those wings yet, and you also don't have the extremely large abdomen, so there's still a lot of development to be doing there. Had a third picture. Okay, let me go back and look, Spectre. Ah, they did. Sorry. Good catch, Spectre. I was so zoomed in, I missed it. This is a cross-section, so you just cut it right down the middle, and just to see what the inside looks like. So really, this portion here is the part that's actually having the developmental region, and then these are all presumably expanded regions. It's probably to allow for air circulation. But again, it's a weird, like, not interior again, because like if you're cutting through, this is where you're hitting the cell barrier of the embryo, and this is just like essentially fuzzies coming out. So it's interesting of why it's that texture again. Nymph looks like the adults. So Supreme Doom, the difficulty here is with the nymph that they haven't aged it. So they never took any samples back to the lab and had them undergo through their regular development. And that, I think, is a limiting factor, right? Because if you aren't age matching them, yes, you can identify that it's a nymph stage. Hi, Kodali, welcome in. You can identify it's a nymph stage, but you don't know where during development it is. So it might be, Supreme, closer to the adult stage than it was to the larval stage. And that could be influencing what we're seeing. They had 2 nymphs, but they were pretty similar. So unfortunately, they're not age matched or even aged in any way. Yeah, so these in the nymph stage are Spectre fly. These are aquatic in that particular stage. Yeah. And yeah, and Optic is right. Like, we don't even know how long it stays in the nymph stage. Also, and this is there's no evidence to suggest that this is true in leaf insects, but there are insects like termites that in their nymph stage will have epigenetic factors acting on them that influence what caste they become. I'm not saying there's castes in leaf insects, but there might be something where there are epigenetic factors acting on the nymph stage that could influence developmental timing, appearance, a bevy of different things. The nymph stage is called the very plastic stage, meaning that it's malleable. Things can still change. Sizes, shapes, again, even casts and behaviors, which is really neat. Pre-leaf appearance stage would be advantageous to acquire. I think, too, Spectre, that when the eggs get activated, it's due to high humidity. And so water retention is always an important factor of these things. They're probably laid near water. And so if you're, the ability to be aquatic would be a beneficial part of that life stage. Here are illustrations of the nymphs. These are really cool. They did 3D illustrations of these animals. And so, if you just look at it, I don't know if y'all can tell based on this view. Hi, Tibbo. Welcome in. The circle thing, a plant. On this image, underneath here, yeah. Underneath there is a plant. Welcome in, Tibbo. So, this looks here, to me, before I had zoomed in like it was just real images. But you can see they went in and actually did a 3D model of these nymph stages. One before. Not sure which one, Tibbo. This one. These are eggs, if these are the ones you're thinking about. These are eggs, Tibbo. These are the eggs for the for these leaf insects. They are distant relatives, Spectre. They're much closer related to stick bugs. So a fair distance away from our mantids. They look like Transformers. They kind of do, right, Ubi? They made like different variations of color based on the sex of the animal. But I thought the illustrations were really neat. These were the freshly hatched nymphs. So these are what they caught in like the egg stage. I'm not sure why they didn't take images. They just made these 3D views of them, but it still is a neat resource, I think. Here, Ubi, they did a bunch of the measurements as well. So here, that description stage. All throughout here is when they're taking all the measurements. Hi, Shark. Shark, I remembered. And Chaos Bales. Chad, if you're not checking out Chaos Bales, leader of the single greatest cults known to mankind. She is a crocheter extraordinaire. An absolutely breathtaking soul is Chaos Bales. Purveyor of the fish hats. And amazing artwork, which I absolutely happen to have. Some Kale's artwork. Whoa, it even disappears. Look at that. How you doing, Queen Kales? Welcome in. Let's go check out Kales. Stone the lamb from the coppers. Run? Or lamb. I mean, either one's fine. Lamb is fine, too. Welcome in, Kales. Welcome in, Shark. We're talking about the new species of the week. New species of the week is, we're talking about seven new species of leaf insect. Purely camouflage, there is a specific survival mechanism other than camouflage. It is, so, Spectre, it is extremely difficult to even test that it is for camouflage. In fact, to me, it's some of the least convincing experiments that you'll ever do. Because the experiments that folks have done in the past is they will put out, like, let's say there was these a recent one that we talked about was these green caterpillars. And these green caterpillars blended in with the leaves quite well. And so to prove that it was for that, they took Play-Doh and they put it into the trees, and they saw if the Play-Doh was shaped like the caterpillar, would birds peck at it? And if they were shaped differently, different color, would birds peck at it? And then they were able to show that, yes, they pecked at the Play-Doh. Which, Spectre, to me, there's so many things, wrong with the experiment, or not ideal, because there's not any olfactory or smell cue, right? It smells like Play-Doh. It doesn't smell like the insect. It's not moving, right? There's a lot of these, like, tactile stimulants that maybe aren't there. And so it's difficult to fully test. So all they're able to really do is that they can hypothesize that it is for camouflage, because it does look like a leaf. But actually testing that, to me, and this is my version of how you would do that test, you would have to mutate the animals to make them not look like leaves, or even change their structure a little bit. Now that means you have to know a lot about their developmental biology, right? So you can do that, and then if you can change their shape and body plan, and then they get eaten by birds versus the ones that don't have their shape changed, don't get eaten by birds, that might be a beginning step to prove that it is in fact the reason they look that way is for camouflage. But you'd have to have a ton of different experiments to test that. Even better before it's been re-blued and re-purple. Ooh, Cal's Bells, I bet it's beautiful. Ma'am, you do have some of the best hair on Twitch. There is another gentleman, though, Kels, by the name of... Well, I say another because I would say my hair is pretty nice. But our friend Plain Old Trey... Kels, Plain Old Trey quite possibly might have the best hair in the streaming business. I know, Kels, I'm throwing down the gauntlet here. And you might be like, Belint, that's madness. How dare anyone have better hair than me? But... Kels, you check out our friend Plain Old Trey. Amazing gamer. And he's got amazing hair as well. Tarzan! How you doing, Tarzan? Yes, Tarzan. New species of the week, leaf insect. You have seen it, Kels? I love your face. You're so kind, Kels. So different light detection, sense detection, air taste, electromagnetic sensory systems. Yeah, oh, for sure. Spectraflight, we have a genetic sequence, but we have not mutated anything in these animals. So you'll have no idea. Like, you can predict what the genes do, but the genes that turn you into a leaf-like structure, you don't fully know until you actually break it. You have to break that gene, and then if the gene is broken and you have them look like a leaf or not, then you'll know what the rule is for it. Is the reason they haven't? Money. Honestly, Spectraflight, it's all about coin. If there is no funding available for this kind of question because it's not directly related to human health, then that's why. Yeah. Hi, Tarzan! T-Bow, I'm doing alright. How are you doing tonight? I got the little one on the mic here to make sure that if she gets fussy, I can run on over and check on her. How come we have no Groot? Because trees cannot yet move. We do not have that tech, Spectraflight. Trees, the way that absorption works for them have to currently be stationary of how they acquire their minerals. Trees move, Spectre, but only insofar as usually light taxes. So they move towards light. But and that's visible, like if you were to have something like an onion, like a green onion. If you put that in some soil and you shift around your lamp, it will start growing towards that particular lamp site. There is that kind of movement, but not from the soil. Yeah, at least not at this stage. Not at this stage. Okay. So here are the measurements, Ubi, that you were asking about. So Ubi, if you're here, my good sir, here they measured the different species, right? And it's they're looking at like the length of body and 7.87 millimeters, 78 millimeters, almost 79 millimeters in length, for example. So that's your rough measurements is where you're getting into. And what's nice is they measured like all their holotype females, the males. They did the antennal segment counting as well. And I believe in citing this figure. Where's figure 5? Spectre, if it's super unrelated, I may or may not know the answer. So in this one, Ubi, is where they're marking what each segment is that they're measuring. So like this, they have the scale bar, that's 25 millimeters, and then they checked out different body part regions, and so they're measuring like leg length, wing height, basically a very large number of measurements that you're able to take. One of these measurements that I was actually doing too on our leaf cutter ants was even like width of the eye. So if you zoom in here on the head, Ubi, the measurement from the edge of the eye to the tip of the eye is a standard one that's taken. And then also downward and then also across. So you get 3 measurements for the eye, and that's before you even do anything deeper counting. So like one thing you also do in terms of counting is the res is not good enough for this image, but remember on the eye there's eye cells called ommatidia, and you can count each one of those. Right, and you can count every single one of those eye cells, like each ommatidium singular, and that is a measurement that's also then later used. That's a new one that you're able to add in, and that's relatively new because of the resolution of cameras, which I think is a neat one. Grass, when injured, releases a special element to warn other grass. Yes. Measure that electromagnetically. No. Spectre, they have done auditory cues, and if you look, we have a TikTok on it where we took a clip of when we were explaining it. It's actually Bit Gamey's sound alert plays into it. Guys, Creatrix Brit is here. If you do not know Creatrix Brit, she is one of the single greatest 3D printing educators and creators, not just on Twitch, but the entire world. I've had the pleasure of having one hour session talking with Brit, and I've learned more about 3D printing in that 1 hour than I had across 30 plus years of life. Please go follow Creatrix Brit, a wizarding Sith Lord, if you will. So if you take Voldemort, but not beaten by a high school student, you know, like much more of a like galactic ruler, but in a wizarding world kind of feel to it, right? Love your face, Brit, ma'am. I hope you had a great day today. Welcome the heck in. Talk about this new species of leaf insect that was discovered. No, Spectre Flight. They are closely related to stick bugs, of all things. Very close relative of the stick bug. So I actually have a couple of videos on these animals. I do have another image of them. Let's see. I think that's the last. Oh no, there's a ton of figures. I forgot how many there are. There are not now. There were witches ages ago, Smikes. In fact, Harry Voldemort's. Because you have hair, Brit, like you have stunning hair. Is that why, Brit? I'm fine with that. I'm totally okay with that, Brit. So yeah, they have more images of like each of the samples that they identified. Remember, they found 7 different species, and so they just have images of every species. You know, dorsal, ventral, side view, antennal view. Basically, the images that they were using to take the measurements, they put into this paper as a figure. Here is another one of the egg capsules. This is a different species. And this is what its egg capsule looks like. And again, there looks like there's little hooks on the egg. But there's no yet functional reason as to why it looks like this. At least they haven't proven anything. Again, you'd have to do genetic tricks on something to prove that, but given that they just now have the sequence, they're not going to be able to do something like that. Not yet, anyway. Grimly had asked earlier today where these animals were located, and so here they do. So here are some of the species. So here's 4 of them. There's 3 on the other end of the figure, and they actually show with these colored shapes where they found them. Kels, I love your face, man. Birds, small mammals, Spectre, also other insects will also eat them. Things like ants will also eat leaf bugs. So a lot of different creatures will actually end up eating them if given the opportunity. Like a lot of opportunistic animals. But let's take a look at some of these leaf insects. Here are some big ones that I think are quite cool just to be able to see in action. go. Well, Spectre, that's what we were answering a little bit earlier tonight, right? Is it you were asking about the experiments that they've done, if it's worked or not and proven, and we were saying that they have been tested with using Play-Doh and other insects, but not definitively shown one way or another to actually work, because these studies are really difficult to prove that. Kells, most of you, how dare? Raiders! Spleen! Be gone. Raiders!
SciAnts stream, but we ain't again, we over here buffalo. You be checking what we got. The trucks arrived. I promise that I have prescribed you vibes. The vibes we got are giant. Shout out to the stream for the raid. That's SciAnts.
Raiders! Welcome on in. Spleen be gone. First time chatter, welcome on in. Mechaform Jack Frost, hello, hello. You were told to bring everyone here. Spleen, thank you for bringing everyone here. How was your stream? What were you making and crafting? Tell me all the things. I want to know. We love our makers and crafters here. What were you up to? What were you doing? What can I share with the party people? I have your Instagram already pulled up and ready to show off. But if there's anything else that we can share via any kind of link, I can share with your TikTok as well. We'd like to know what you're working on today. You can feel free to drop a link into the chat of tonight's projects or into our Discord. We have an art section. Anything is Gucci. I'd love to see it. It's going very well. I was working on a little hammer built on a metal lathe today. Very nice. What size? How little of a hammer are we talking? How little of a hammer are we talking? Tell me. But Spleen, welcome on, and guys, go follow our friend Spleen Be Gone. I'm going to show off your stuff in just a moment, my friend. I'm going to quickly introduce myself. My name is Balint. I'm 1/3 of SciAnts streams. Other 2/3 are my lovely wife, Lita, and our daughter, baby Alona, who is currently on the baby monitor. Lita and I are research scientists, and we do interactive science streams here on Twitch most days of the week. We do microscopy, art, sci-fi, science news of the week, deep dive scientific topics, games, 3D printing, IRL streams, and more. If that sounds like it's up your alley, consider hitting a follow button. Tonight we're talking about the science news of the week. In particular, we're highlighting a brand new species of leaf insect that was discovered in the Southwest Asia this past month. Raiders, welcome on in. Before we continue on our topic, I want to show off some of Spleen Be Gone's pieces. So here is the Instagram link. Here is that's the TikTok link for y'all. Go drop Spleen Be Gone a follow there as well. It takes 100 followers to get you to have the link be part of it, so make sure you go drop them a follow. Big enough to cause a knee-jerk reaction, says Mechaform. 1 inch round, 2 inch long on the head. Nice. Spleen, what is the purpose of the hammer that you've built tonight? See, making rigs, new shop tables, pineapple twist, antique metal lathe. So a lot of build vintage pieces I see going on in there. Much on TikTok. That's okay. Spleen! I like to show off all the things. Spleen Be Gone. I already have the Instagram as well. Here I'll go is the Instagram page for the lovely Spleen Be Gone, aka Build Forge Paint. Let's see some of these here. See, that's the same piece. I found it, Spleen. It's the same one as the TikTok video. Looking at the antique pieces here. Ooh, custom jewelry that you make. These are lovely. Oh, is this like a Damascus steel teardrop pattern inside of the ring? It's actually wood on the inside. Nice. That looks really cool. I recently purchased the lathe and have been rebuilding it. The hammer is just me learning how it works. Nice. Hammered steel on the outside. Lovely. This is... You've done beautiful work in this. Like... That patterning on the inside is really, really lovely. And I like even like the texture on the outside too. Like, it must be a really tiny hammer, right, that you're hitting it with. That is really, really pretty. Oh, and look at this one too. Another ring, stainless steel, eucalyptus wood. Why eucalyptus wood, Spleen? Very pretty. Curious as to why that wood selection. Check these out. Rune Worry Stones. Nice. Very lovely. And a piece of it from another project. Oh, cool. Yeah, no, I like that. It turned out lovely. I didn't know if there was some, like, blacksmithery reason for using, like, that kind of wood that would be, like, work well with a particular metal. We have some blacksmith friends here on Twitch, and I'm always asking, like, oh, why exactly are you doing it like that? Well, it's sometimes just like, well, it's pretty. Like, oh, okay, that's cool. This is always a cool shot. Etsy orders, very nice. A weapon, a new mace. Spleen, you have maces? Very nice. Super cool. Yeah, these turned out great. Oh, wow, look at the candles. Similar kind of texture to the ring, right? Jack-o'-lantern shows the original mace. Okay, hold on. Oh, nice. That's excellent. Well done. That's a great hit right there. Very cool. Guys, please go follow our new friend Spleen. Spleen, thank you so much for raiding on over. Guys, oh, I already did the Fancy Pants shout out. Let me do the classic shout out. Here we go. It's on an hour delay. I apologize for that. Spleen, thank you for the follow as well. Guys, go drop our new friendo a follow. We love our makers and crafters here. We do artwork here. Digital art usually of 2D or 3D. We make our own emotes and our own animations in some kind of scientific context. Sometimes it's just silly, like this.
The hands of death could not defeat me. The sisters of fate could not hold me. And you will not see the end of this day. I will have my revenge.
Why we do this kind of thing? It's to excite people about model organisms, so organisms that are studied in the research lab. If they look something like the real insect, it's kind of, people will have like a negative reaction to it because they can be grossed out by them, or they kind of look scary. But if you see an ant that looks like Kratos from God of War, or like a character from Marvel, it's not as scary. And so maybe it's a little bit more approachable and you're able to ask questions about it. So we do science art for that. We do microscopy, scientific topics, science news, IRL streams, and more. And we're glad to have y'all here. Again, welcome on in, and thank you so very much for the raid. Those of y'all, again, coming in on the raid. We are talking tonight about these leaf bugs. Is our paper so far, this brand new species of animal. And we're about to watch a video on leaf bugs. And just of how they look, and again, a little bit about their behavior.
When is a leaf not a leaf? When it's got a face and 6 pointy legs. It's a giant Malaysian leaf insect.
And so this is a one from that area where it was discovered. And yeah, Spleen, there were 7 new species discovered in this paper. And what's wild is apparently a lot of them look alike, where morphology measurements will not tell you that they're different species. You have to do the genetic sequencing. So people had thought that these animals were, like, there was like 2 species in India, but it turns out they have at least 4, and that's just what they actually ended up sequencing for it. So Spectre, we did look at a map where they were discovered, and they were discovered, the paper was published in August for these 7 new species. And I'll put the paper on Discord, as we always do, so y'all can take a look at the paper and the figures if you want to see.
You are what you eat definitely applies here. They blend in with their food, which is also their home, to hide from hungry predators.
And like we talked about earlier, that's not like definitively proven because it's very difficult to prove that kind of hypothesis and or question. But it's a good suggestion. Yeah.
But how hard is it to be a leaf, really? You just stay still. Really still. But it takes more than that. The mimicry is masterful.
One of my favorite parts, again, of these is the wing veins also mimicking the leaf veins. So you can see how it looks like it's the entire vasculature system of a leaf, but imposed on the wing of the animal.
Their bodies have intricate veins, just like a plant does. And on the edges, brown spots, like a crinkly, damaged leaf.
I feel like is next level camouflage.
Type of camouflage, where you copy your natural surroundings, is known as protective resemblance.
They even have that little bit of sway movement, right, that a leaf will have if there's wind, and it's like nailing it to a T on the mimicry.
It's all they have, since their short antennae and small eyes don't really help them spot predators. They cling there with tiny bifurcated toes. Moving is the biggest mistake this insect could make. Even when it's time to lay eggs, it barely moves a muscle. The eggs just plop down to the forest floor. They're camouflaged too. They look like teeny brown seeds, or leaf insect poop. That's an egg, and that's poop. The active young nymphs start out brown and transform gradually, taking on a little more green over time. This one's halfway to a fresh leaf. Its color is set though, it can't change it on the fly. But this nymph still has a big migration to make, getting into a tree undetected. So, it sways. Yep, like a leaf in the breeze.
Noki, I had a chuckle about that too. Hi, Derpington, by the way. Welcome in.
That funky little walk is all leaf, a dance of disguise.
Just look at the size difference too. Right, there you have the adult, and there you have a nymph. So there's still how much growth it will undergo, and there does mean there's going to be adult molts on this. So we've talked about there's insects that do molting, like these, and there's ones that don't molt, for example, like your ants and your bees. Just an interesting aside.
It finally ascends, it'll settle in seamlessly, like this adult. Between nymph and adulthood, it molts, or sheds its exoskeleton, 7 times. Its appearance ages just like a leaf, its brown spots getting bigger. It may never move.
Which I think is super cool that, like, that damaged look to it will increase as it ages. Guys, I'm going to take a quick BRB. Baby Alona is being fussy. I'll be right on back. Give me a shake of a leg of like 2 minutes. Oh, I hate this poop shot.
Very sophisticated, yes.
Mocha, one my. Hi, Yogurt Carol!
See your butt.
Well, that's for the Antonia butt wiggle, you know. That offspring can breed as well. And what's ah! Coffee!
Cracks at them, and it.
There's no underpants worn by these animals. Every other name we go, you know, like we have Dougie, Jern, Barty, Jern, Claire, Jern, Date Night In, Jern, Big Gaming, Jern, 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 Jern. Jern, 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 Jern.
Surely, you must be thinking. There's something Australian.
And don't call me Shirley. It's like, oh!
Neater, 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 neater.
PG fan. PG fan.
See, I need. One more time.
A DVD worm
No, I don't get
instead
it.
of a tapeworm. Cassette tape. Why is it hot?
Because I didn't want it cold. Listen, if a girl's gonna make a super virus, a girl's gonna make a super virus.
Dueling Needles and Crazy Mama 88 Coffee Rock. Grimly, Sir Grimly, thank you for the 1,500 biddies. Thank you, Grimly. Guys, I am so sorry. She threw her pacifier. That. Andrew, thank you for the 200 biddies. Andrew, yes, I did say shake. I really did say shake a leg. I apologize, guys. Thank y'all for the bits. Little one threw her her pacifier down, and it fell like between the bed and her like surrounding cover. Maelstrom and Spectre for the biddies kicking off a hype train. Y'all, we wildin' tonight. We wildin'. Nice. Thank you for kicking off that hype train. Already at level 2? That means we have a raffle to do. That means, y'all, we already have a raffle to do. Let me set up the raffle. Andrew! Andrew, thank you for the 5 Tier 1 gift subs, Andrew, to the Dinosaur Dave, Commander Falcon, Zemfaal, I'm Only a Big Child, and Nin Player 89. Thank you so much, Andrew. My goodness gracious. Inspector, thank you for the biddy. Spectre Fly. Y'all, thank you for that. Let me go ahead and set up a raffle. Okay, it is live, ! Raffle, and what you can win. Hold on. Stickers and or magnets. Spectre, thank you for the 99 biddies, Spectre Flight. These are what you can win. Stickers and or magnets. There are going to be at least 2 of these. Every level of a hype train complete, we do additional raffles. Complete a level 5, and we'll do raffles for our sticker sheets, hard enamel pins, as well as I'll put on a ferret onesie, but absolutely no pressure to get us there. Hello, Mary Ann. Welcome on in. And yeah, we'll just keep going until the time runs out, and then we'll finish up our leaf insect. And then we actually probably go on to the next topic, which is, I think we're going to talk a little bit about aging next. Spectre, thank you for the 140 biddies! Spectre Flight! Thank you, Spectre, for those biddies. Yes, guys, ! Raffle to enter. If you do win, I do need your mailing address. So, Shipwreck, I noticed don't think I have yours yet. So our friend Shipwreck from earlier, if you can send me your raffle winning address so we can have it sent off to you, as well as to anyone else who will win tonight, I will need that mailing address so we can get this shipped on out to you. And if you do spam it like Spectre is, you might just get removed. Nightbot might have a setting on it that says if there's a spammy thing on there, then it removes you. Spectre, I don't know what to tell you. I have the anti-spam enabled. I have it enabled. I don't know why, Spectre. I think it just makes me chuckle ever so slightly. See? Andrew's like, yep, it does have a spam filter. Andrew's flexing that sword, chat. Guys, go follow our sword flexing guru that is built like a Greco-Roman god, the lovely Andrew CZ, folks. Technical wizard extraordinaire. Go check out Andrew. Shark, feel free to enter, though, if you would like to. Absolutely no problem at all. Exclamation point raffle to enter. Y'all, thank you again for kicking off another hype train. Far, far, far too kind. Far too kind. And we'll continue on. Whoa, my hair needs to be haircutted. Eventually, we'll get haircutted, y'all. Will it be tomorrow? Maybe. Possibly. Will it be before Sunday's stream? May very well be. Tomorrow, we don't have a stream, y'all. But we do have... No, Spectre. I cannot look like a savage. We do not have a stream tomorrow, but we are having a Discord watch party of the movie The Meg, starring Jason Statham. I will not have that long hair, Spectre. I refuse to have that long of hair. This is already making me uncomfortable, but I have not been able to go because of work and or Ilona and or Lita. So at some point soon, I will be going to get haircutted. Absolutely, without question. Plus, I can't have Andrew be upset with me. You know, I have to have a nice haircut for when I see Thug Shells again. It's just, and Andrew, both of them, in fact, have the nice haircut for that. It's a whole thing. Okay, so tomorrow we will be watching The Meg. So Spleen Be Gone, tomorrow we're watching it just like empty, mindless enjoyment. But then next Thursday, we'll dissect the science behind The Meg. Yeah. Scheduled for the 5th of October. Gotcha, Andrew. Very nice. So yeah, Spleen, we do deep dives usually on Thursday nights, so we'll do a Thursday night deep dive on the science behind what we know about the Megalodon, and then why the movie is silly. We've done deep dives like this on others, like more science-y movies, but nothing that's quite out there. But people voted, and we're like, okay, so we'll go ahead and watch it tomorrow. Guys, 30 seconds left. On the hype train, exclamation point raffle to get up on in there. If you win, I do need your mailing address. If we don't get your mailing address, then I am unable to ship out your items. Which I think goes without saying. But, just so y'all are aware, that is a component of it. That is a component. Hi, Zeddy!
Hello there.
Guys, go follow the amazing, the breathtaking, looking cool Zedman, the coolest looking Zed head in the entire world. If you aren't already, Pokemon streamer, card collector extraordinaire, all things fancy. Please go follow the one, the only legendary looking cool Zedman. How you doing, Zeddy? All right, let's go ahead. Now that Zedman's here, let's go ahead and run this raffle in 3, in 2, in 1. Roll it. Marianne. Marianne, you have won a sticker and or a magnet. Marianne, what would you like? What would you like, Marianne? What would you like, Marianne? Of these, so 1 through 13 are stickers, 14, 15, 16, and 17 are magnets. What would you like, Marianne, off the menu? What are you feeling? Magnet 14, you got it, Marianne. Marianne, could you please DM me on Discord or Twitch Whisper a mailing address? And we will ship it out to you ASAP. Little one and I might ship it out tomorrow morning as well. We're going to the farmer's market when she wakes up and has her breakfast, and then we'll try to drop it off then, if not afterwards. All right, one more raffle. In 3, in 2, in 1. Spectre Flight. Spectre Flight, come on up to the front of the line. The naughty Spectre Flight, the purveyor of chaos, has won a sticker and or magnet. Win Ant? You don't want it, Spectre. All right, so I'm going to re-roll it. Is that okay, Spectre Flight? I'm going to re-roll it, Spectre. Spectre, we Gucci on a reroll? Please re-raffle it. You got it. In 3, in 2, in 1. Better yet, Shark. Better yet, Shark, you have won a sticker and or magnet. Better yet, Shark, from the kindness of Spectrefly, who passed on his winning. What would you like? Better yet, Shark. Better yet, what would you like? What would you like? You got stickers of 1 through 13, and then you have 14, 15, 16, and 17 are magnets. Shark. The lovely Shark. What would you like, Shark? What would you like off the menu? There's no secret menu yet, but there might eventually one day be a secret menu too. Hello, Supernatural Rider. Welcome in. 14, you got it. Partner magnet it is. Alright, Shark and Mary Ann, please send me your mailing address. You can either send me a Twitch Whisper or a Discord DM. I see Shipwreck has sent me one. Mary Ann has sent me one. Better yet, if you could also send me, it can be on Discord or Twitch Whisper. Either is fine, just to make sure it comes on in, and then I'll try to package it up for it today. There is no secret merch that exists. You know, probably not yet, Andrew. Not yet. But there eventually might be, possibly, one day, potentially. Guys, go check out lovely Supernatural Writer as well. Co-working streamer, writing streamer extraordinaire. My friend Chelsea says you are a handsome science man. Your friend Chelsea is lying. Gwen, no one has ever said that I was handsome. Lita says this, but I think she says it just out of jokingness. Gwen, the one woman that has commented on my looks was in middle school, and she said I was ugly. It's true. It was at a middle school dance, Gwendolyn P. I went to the dance.
Ball ain't the science guy. Bailing the science guy.
SciAnts rules.
Bailing the science guy.
SciAnts is a partner on Twitch. With lovely wife Lita.
Bailing the science guy.
Well done, Big Gamey. Hi, Portal guys. Welcome in. Hi, Tiffany. Welcome.
Hey, Belen. It's time to say something nice about yourself.
Thank you. Male, Big Gamey's a little naughty. So, as Big Gamey interrupted me, Gwen, I did go to the dance, and I asked the girl to dance with me, and she told me no.
Look at its little feetsies!
Because I'm ugly. And so, Gwen, that's the real truth. Your friend Chelsea is being a kind, sweet soul. Just like our friend Big Gamey. Guys, go check out Big Gaming. Legendary game developer extraordinaire working in the Unity game engine, making a video game called Homeward Hound, A Dog's Life Battle Royale. It's amazing and breathtaking, in fact. And he is also the single greatest father in the world. This man knows no bounds of how amazing of a father. And the thing is, he tries to tell me that, oh no, he's just doing like what any dad would do. That is the biggest load of malarkey I've ever heard. Bit Gamey is, if anyone had a father like Bit Gamey, you would never, if everyone had a father like Bit Gamey, no one would have daddy issues. In fact, no one would ever feel alone in the world, because this man is always there for his children and his wife. He is, Zed, top tier human. I don't know how, what BitGamey underwent to become such an amazing soul. But whatever happened in your life, BitGamey, that made you the way you are today, please write an instructional manual. Please put it out there, and let's make it mandatory reading across every grade level. I mean, seriously, if there were more people like you, there would be no war, plague, famine, anything in the world. It's just a whole thing. It's just a whole thing. No, Spectre. No. I'm, by number 1 of Bullet, you mean number 411. Debs, how are you doing tonight? Grimly, I would also buy that, but I would have to travel down to see BitGamey and have him sign it. Man, how dare! BitGamey, thank you for the 100 biddies. And yes, that meant everyone in this chat. I love y'all so hard. Tired?
Ah! You are wholesome as ferret. Yay!
Debs, I've honestly been worried about you, but I get nervous in reaching out because Debs will usually send me a message that says like vibe check, like how's it going, and I haven't gotten one for a few weeks, and I'm always like, I hope Debs is okay, but then I don't want to seem like overbearing of a friend and like ask, and so then I'm always just like worried, and then I like last night I was laying in bed, I'm like, God, I hope she's okay. And I'm like, no, Andrew would tell me if something was wrong. And then I'm like, no, but I can't bother Andrew about how Debs is because that's even more overbearing of a thing. And so it just started spiraling. Debs, but I'm glad you're okay. I'm sorry you're tired, but I'm glad you're okay. I know that was like a very like long journey I just took you on about like the insanity in my brain. Stress about blood exam, but all good. Okay, as long as you're all good, Debs. I'm sorry about the stress and the tiredness, madame. I love your face. Debs is our lead content creator on the YouTubes, and she's been absolutely like redonkulously crushing it lately, as she of course has been. But like, my God,
you are wholesome as ferret. Yay!
So go check out the YouTube's page. Copper is high. Be careful, Debs. All kinds of metals, you have to be careful. And of course, like the heaviest metals, like Metallica and Anthrax and Slayer and Megadeth. Yeah, the big four. Yeah, you don't want those big four too much in your system. In your ears, yes, but otherwise, you're all kinds of Gucci. So, Andrew, I would argue Godsmack is more hard rock than metal. And I would actually argue nowadays, even the big four. Of the big four, I mean, Slayer has retired, but Slayer would have been the only one that I would have kept in metal. And because nowadays metal is more screaming, and like even Sabbath nowadays, Supernatural, I would put more in the hard rock category. A lot of the metal has been taken over by speed metal and scream metal and black metal. And it's just like your classic metal definition isn't so much there. The new metal, Andrew, has also shifted, right? Because Papa Roach and Disturbed were considered new metal for a while, but now I'd also argue that they've shifted right onto hard rock. Or it's maybe the spectrum is just shifting of like maybe at that point that hard rock would have been metal in the 90s, but now what we're getting used to, I don't know. It's in my veins of it. You'll send me some albums. So Zeddy, I will say the straight up screaming I don't get, because I need to be able to understand what they're saying. But something like Hi Travel the World, welcome in, madame. How you doing, Travel the World? Something like Slipknot, where you have screaming, but then the amazing vocal range of Corey Taylor, like off his Stone Sour stuff, also off the his solo stuff. That mixed together is really, really cool. But I don't like the straight up only screaming. I know it's hard rock and metal is the reason I can't hear anymore. That's a good reason, Shark. I mean, that's the reason why my hearing isn't as great as it used to. Apparently, Shark, if you hear ringing in your ears, it is those cells dying, and you're hearing that note for the last time. Opposite of Screamo Band. But BitGamey, they were British, right? So it was like at least cultured. Five Finger Death... So I would call Five Finger Death Punch hard rock. I would call System of a Down hard rock. And I would call Nine Inch Nails classic rock. At this point. In this moment is great. Supernatural writer. Their lead single that starts with the W that I shouldn't say is one of my favorites of theirs. Look, 9 Inch Nails at this point, Smikes, all their good stuff is now classic. Right? I have to say that your modern 9 Inch Nails, like the copy of a copy of album, I'm just pretending that didn't even exist. Ginger and Spirit Box. Zeddy, what was the oh yeah, Metallica's 100% classic rock then. It's played on classic rock stations, without question. Kiss is classic rock, yeah. AC/DC, Motley Crue, yeah. All have gone into that one. It's gone. Listen, Debs, because I got nervous, and we can't just make sure that isn't. No. Have you heard "Copy of a Copy of"? It's terrible, Smikes. It's terrible. He's a very talented musician, but I do not enjoy any of the new stuff. Poison, I've seen Poison too. Actually, Supernatural. I saw. Oh gosh, who is the lead singer of Poison? Bret Michaels. When I saw Poison and Motley Crue, the tour was called Glamageddon. Okay? And Poison was about to come on. And there was a guy that came into the crowd. And he was dressed like Bret Michaels. And he started signing people. Okay? He was signing women and men's body parts. And cell phones. This guy was walking and just signing everything. And then, Supernatural writer, the real Poison came on stage with the real Bret Michaels. Supernatural writer, someone in the crowd where we were looked at the fake one and goes, "Get him!" And they started chasing after him. He got away. He jumped and he ran. But no, this was not a Bulent dream. I have a picture somewhere of fake Bret Michaels at this concert. I think it's on actually Facebook. It's on one of the Facebook albums. I do have that picture. But did you get home safely, Quoth the Raven of all? Quoth, are you home safely, Quoth? But Gwen, he had the hairband, like the do-rag kind of thing. He had the facial hair. He had the blonde hair. He had the eyeliner. He had like the leather clothing. Like this dude looked like Bret Michaels. And even we were a little ways away from him, but I looked over and I was like, that looks like Bret Michaels. So Gwen, it was a very convincing Bret Michael. Until the real one came on stage. And I just remember someone, "Get him!" And this was down in Georgia. Of course, he just hopped the rail and ran away. And, then no one really ran after him. But you did make it on excellent, Quoth. Quoth, we may have derailed a little bit. Talking about Bret Michaels being, fake Bret Michaels being chased. Look, Quoth, he was signing people's wives. Okay? Fake Bret Michaels was signing people's wives. Of course he was going to get chased. I also saw him signing cell phones, which, like cell phone cases and everything. I'm like, God, like this guy. Yeah, don't tell my wife. That's the lovely geisha. Oh man, that from Argentina, end of every joke, it would finish with "Don't tell my wife." Made no sense whatsoever. Except the final time he said it it actually made sense when he went to celebrate like him getting a professor position. But like beyond that, signing out at every end of every email, "I'll tell my wife." All right, I don't know why. I don't know why. The Cure, I think it's also classic at this stage. Super. I would argue anything on a classic rock station now is classic rock. I mean, most of Metallica is classic rock. Guns N' Roses, like. Heck, at this point, 90s and early 2000s Disturbed and Godsmack are also there. Yeah. Did he do his job? The fake Bret Michaels? He ran away. He ran off. The real one did a great job, singing. It's Friday, we're in love. That's true. Gotta get down on Friday. Huge difference between Nirvana. There is. There The new Sabbath is almost like, I don't know if you heard the brand new Sabbath. I would almost call that light rock at this point. Even like the new Ozzy, like the Scream album of what was that, 06. You know, POD at this point is also classic. Like it has a bunch of classic elements to it, right? I mean, don't get me wrong, Super. I like these bands that we're listing. It's just either they have, even like their new stuff might be considered hard rock or something, but then they have at least a chunk of their catalog that's old. Lita made a new animation that's not live yet because she hasn't exported it yet for me. But the throwing of the tomatoes, we're doing a seasonal one where it's going to be throwing of jack-o'-lanterns. Yes, we do need to do it. And Grimly, it'll be so much anger at me. Guaranteed. Listen to too much metal? No, you don't. Zeddy, did you know that there is a metal electric violinist? I forget her name, but she is incredible on TikTok. And she does covers of pretty much anything you want on TikTok. And then she's on tour right now with an electric bagpipist. Yeah, I've sent you her videos. She's on tour right now with an electric bagpiper. Yeah. And she was on stage like with some big European rock band about 2 weeks ago. They brought her up on stage to perform with them, and she was like decked out in spiky metal. Like it was, that woman is so epically talented on that electric violin. And then the electric bagpipist chick too is just like, they put out actually a music video today or yesterday. They covered Freebird. And you might be like, Blant, they covered Freebird? How dare? No. They covered Freebird, and it is like leveled up a Freebird. Like, they did a great job. I think of Wonder Woman. Yes, and actually, Grimly, the Wonder Woman animation that we have on screen, like with the ant, the sound I actually took as that woman doing the electric violin of the Wonder Woman theme, like from the movies. She did a cover on her violin, like again, like the electric version, and that's what we were playing. Like that's what's playing over the animation, because I just like it more than the original version of it. If you, we can, all right, let's.
In dreams, one is not
Big Amy!
by Earth
Hi there.
limitations.
Sleep
What
well,
does
Big
that
Amy.
mean? Come.
Sleep well. Thank you for the science guy redeem right at this. Like Gwen's friend, like, hey, I think you're handsome. No, I've been called very. Big Game's like, we will not have that here. Oh, listen, Grimly, listen. That cover debate, I am 100% on board with you. However, here's where I get controversial, Grimly. I don't like the Beatles. And in particular, I appreciate them. They did very important for the world of rock. But I do not like them. In particular, I do not like the vocals. I do not like anything about the vocals. Okay? But Godsmack covered Come Together. And I'm all about that cover. And again, it's the lead singer of Godsmack doing the vocals that gets me. They also covered Metallica's Nothing Else Matters only on a piano. They didn't try to out Metallica, and I really like the piano version. Now, the craziest version, which maybe Zedman knows about this one, there is Mama Said Knock You Out by LL Cool J, is covered by Five Finger Death Punch. I heard that cover before I heard the LL Cool J song. And I was floored when I heard the LL Cool J version. Because the LL Cool J version's like, I'm gonna knock you out. Like, it's like this, like, you know? It's like all kinds of loosey-goosey. And then the Five Finger Death Punch one is like, I'm gonna rip your throat out. Kind of, I'm gonna knock you out. Like, it is the most aggressive feeling song. It's nothing at all similar. It's madness. It's absolute madness. Quaff's Falling in Reverse did a cover up. Oh, which is better than... Ooh, Quaff. That might be fighting words. Lita's not here, Siraguchi. Black Sabbath spawned a complete genre of music, stoner rock, and it sounds more Sabbath than Sabbath. Didn't Cliff, the guitarist of Sabbath, he founded a band, Black Label Society, right? And that is a pretty Sabbathy style. Zach Wild. Zach Wild started Black Label Society again after he left, like with Ozzy, and that feels very much like that. Who is Randy Rhoads, Mikes? And is this recent? Disturbed, yeah, Land of Confusion, Shout. The one Zedman too that's been tearing up the charts is Sound of Silence. I think they did a great job on that cover. There was, I think, one on the new album too, but I forget which cover song that was. Oh, it was Ozzy's guitarist when he was... but no, it wasn't just Randy Rhoads, because didn't... Zach was also his guitarist, too. Zach Wild was also in... Okay, gotcha. Was both. Gotcha. Okay, gotcha. Beartooth, I don't like super. Because of the vocals. I don't know... Super, it's not a good reason. But the vocalists, like, turn me off. Another good vocalist, though, is... Zeddy will agree with me on this, is Trivium. Trivium's another one of those very few rare ones that can do screaming and also melody, which is really nice to have those two differences. Sabbath was always Tony. Okay, so it was only in the Ozzy band. Gotcha, Cliff. Was more famous, really? That's the first time I've heard that take. I've always heard Zach was more favorite. And more better known. Newer Beartooth, my got into in the past few years. Trivium is great. We saw them on live. They opened for Godsmack and Stained one tour, which was really, really cool to see that. It was the In Tears album. Quoth appearance Sound of Silence live is like tear jerking. Quoth, I've seen Disturbed live, and it was very disappointing. It is, and I never thought I would say this of a band that because I love Disturbed, like top 5 bands of all time for me. They overstuff their set. There is too much in their set. Like, there were no gaps in between the songs, and they also didn't elongate any of the songs, so like you didn't really get any audience participation singing along of their music. And so it was just like boom, 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 boom. So I felt too mechanical, and it was weird because I really thought, like, you know, if it's more songs, like, that should be great, but it felt just like, again, not enough with the audience, just too over stuff, like vocals, and all the audio was fine. It just didn't feel like as much of a live experience. Korn puts on a great show. I saw them when they put out their dubstep album. I haven't seen Rammstein, but they sold out the Atlanta Coliseum, and we weren't able to get tickets. Best show still I've ever seen, I know I'm biased, is Godsmack. Single greatest band show I've ever seen. Followed up, Papa Roach puts on a great show. Like, the smaller band, but absolutely amazing. Everything's on fire all the time. That's how Godsmack is, but like, well, I guess not all the time. It's select when it's on, but when it's on, it's like everything's in flames, and the way their light show works and the drum battles that you have, it's really, really good. Bon Jovi also puts on a really good show. I've seen them a couple of times. I don't know if there's anything like really terrible that we've seen. Skillet, do y'all know Skillet? Skillet puts on a really good show. Black Label Society, Cliff, put their show, I near fell asleep. That's fair. Trans-Siberian Orchestra is always, that is true, that is always fire. Voodoo, it's all amazing, Zeddy. You can't. You know what's boring though, Zedman? That I was disappointed in? Slayer. I about took a nap during Slayer. And I love screaming the word Slayer. I really don't much care for their music, but I was like, Slayer! And then it was just so sleepy. Colombian grindcore band named Purulent in the 90s. They have a song, scares the heck out of me to this day. Oh no. Gwen, there is a rap song from the 90s. I forget the band, and I forget the title. I remember the topic. It's a very good rapper, and it was like a 10-minute rap song about a story. And it was played on a debate trip that we went on, and we drove the debate coach drove the school bus, and he plugged his laptop in and played music, and it was very graphic, Gwen. And I remember it was like we were driving to the hotel that night on a Friday from school for the debate tournament, and it was so unsettling that I couldn't sleep that night. And it was like dark when that song started playing, and we were trying to nap like in, the seats of the bus, and I remember it was so upsetting. I have no idea what it is. I have no idea what it's called. I remember the subject matter, which I'm not going to talk about on the stream, or with anyone, in fact. And because I do not need that neurons to like re-trigger of like, I can like vaguely recall it, but I don't want to like bring it back in. But it's the same as like Gwen saying that it scared her. It's to this day I think would still scare me. Yeah. We need to bring out his guitar and demonstrate his chops. I would be terrified, Ubi. Ubi, right now... Actually, Ubi, the amp got damaged. The amp got damaged from the flood. The guitar is fine, but the amp got damaged. So I think... I haven't checked it yet, see, but I know it got like waterlogged during the flood. Maybe it's dried out and it's fine. I'll have to see if it worked or not. Yeah. Anywho, that's our musical derail, y'all. Almost as good as our bread derail. If y'all remember the bread derail. The bread derail was a really fun one. That was a good time. Okay. Are y'all ready to continue on the leafy bug? You ready to continue on the leafy bug? Guys, let's continue on the leafy bug. So, Gwen, Debs, I'm putting a marker. Don't worry. Debs is probably like, oh, good God. The guitar, Ubi, would you like me to bring out the guitar? I will not play it. I don't have it hooked up. I don't know where the picks are. It is not tuned. It is not ready for anything, but I can show it to you, Ubi. I learned today that each sandwich bread is made from 4 lumps of dough. Really? Cliff, is that based on like size or something? All right, Ubi, let me go get the guitar. Ubi, it is super out of tune. It is super out of tune. I just hit it and I'm like, ooh, that sounds terrible. But there it is. I used to have, Ubi, a replica of one that Godsmack used from their first album. It was a really, really cool guitar. But what ended up and this was in senior year of college. And what ended up happening, so Ubi, there was a gentleman who threatened to kill me and Lita. And I had for some reason brought in that guitar to the lab that day. I think it was because I was going over to a friend's to like noodle with the, and he broke it. Like he shattered the heck out of it. So Lita was very sweet. I'm not sure which one it is, Smikes, to be totally honest. Lita got this for me for our first Christmas together. So I've had this since, over a decade now. And yeah, it's a lot of fun, but it is very untuned right now. Like I can hear it that it needs to be like fixed up. I don't know if there's a dinosaur inlay in this, Andrew, but it's nice textured, and I don't know, it works. Again, Ubi, I need to see if the amp needs to be a new one brought. Hi, Luminology. I'm sorry, Andrew. See, it's like that wire is totally off. This one, where it is. Yeah, this one is like totally nerfed. That one needs to be tightened the heck out of because that is not doing anything. Light it off. I do not, Ubi. I do not. When I moved in with Lita the first time, well, like the first time I mean like, because we moved in together in Atlanta, and then she left to go up to Dartmouth, and there you go, Smikes. She left to go up to Dartmouth, and then I moved up to Dartmouth, and then moved into her place. So again, so like we moved in technically twice. First time I had that other guitar, she's like serenade me with something beautiful, and I got so nervous. I was like, no, I can't do it, and then I went and hid. I think like once or twice I might have played her something, but I was mostly too terrified to do anything. Even I was scared to like practice in front of her too, because I'm like, no, she'll think I'm ugly or so. I don't know. Confidence issues, but yes, Ubi, this is the guitar. It needs some work again. Like this, there needs to be tightening up on this guy. Like these are totally off. But anyway, I'll futz with that at some point. I'm still worried about the again, that amp was really cool because my friend Nate got that for me. You only can sing tunes. There's a really cool game called it's not just a video game. You can hook up the guitar to the PlayStation 3, and I have this game called Rocksmith, and it lets you like practice your chords and like practice for songs. It doesn't have a like any kind of plug-in like for a microphone, unfortunately, but that would be a pretty fun way of doing that. Musician, okay, gotcha. I only heard of that Rocksmith one, which is also why the PS3 is still out, even though I don't actively use the PS3 for anything. Like at some point I'm gonna be wanting to use the PS3 to get back into that. I'm like, but I refuse to put it away because I'm like, no. There could be a reason for this. Peter and Paul Mary have an album of folk tunes called Around the Campfire. Oh, cool, Ubi. That is really cool. I have to check that out. But yeah, and Ubi, so Ubi has been asking y'all for over a year for me to even just show off the guitar. And Ubi, you're lucky it's in the upstairs because of the flood. Otherwise, I'd have to go all the way downstairs. But yes, there is the guitar. Yeah, the amp is down on a platform downstairs, and again, I need to see if it works. That amp actually, my best friend Nate gave me that amp because he used to be in a rock band.
In dreams, one is not tethered by earthly limitations.
What does that mean?
Come.
I guess not a rock band. He was part of a punk band, and it was cool because he had that. He had that amp during his time as a musician, and what was really cool is that during the punk shows that he would do, Ubi, there was this guy there that got really angry. But he wasn't angry at the band. He was just like a ragey guy who would always go to their shows. And he would scream at the top of his lungs, "That's really punk!" Every time, okay? And so Ubi, Nate had since retired. He wasn't in the band anymore because he had so many like leg surgeries and foot surgeries that he couldn't stand for a long time, and he just, he had to be done with all of it. But he was like, man, I would really like it if that guy would still follow me. And he like would do that to my experiments. Like when Nate was saying like if he did some kind of DNA extraction. Raiders! Real Fun Studios. welcome on in. Raiders!
SciAnts stream, but we ain't bugging. We over here bubbly. You be chugging what we got. Now the tribes arrived. I promise that I have prescribed you vibes. The vibes we got are giant. Shout out to the stream for the raid. That's science.
Raiders! Welcome on in. Real Fun Studios. Real Fun, how was your stream? What were y'all up to? What were you arting today, Real Fun Studios? Tell me all the fancy pants things that y'all were doing. We are doing some science news of the week today. We have been derailed heavily into talking about my non-existent music career as we pulled out my guitar. Real Fun, how was the stream? What were y'all up to? Tell me all the things. My name is Belint, 1/3 of SciAnts streams. The other 2/3 are my lovely wife Lita and baby Ilona. We're research scientists and we do interactive science streams here on Twitch most days of the week. Microscopy, art, sci-fi science, science news of the week, deep dive scientific topics, games, 3D printing, IRL streams, and more. Raiders, welcome the heck in. Again, Real Fun, how was your stream? What were you all up to? I have your Instagram pulled up and ready to share, but if there's anything else that you want to share with us, you let me know. You can drop it into the chat, pop it into the Discord, anything is Gucci, and we will show off your work. Hi, Music Girl, welcome in. It was great. I was working on a logo and then sketching some ballerinas. Nice! Real Fun. I love the bird sketch with the paint as well. Like, this one turned out so beautiful. There it is. There it is. Look at it. Oh, it looks so good. No pics yet. No worries, Real Fun. We can look at the older stuff, right? Guys, Real Fun Studios, go drop them a follow. And here is the Instagram page as well from Real Fun. You can check out some of their additional artwork. These were, what was the piece that we were like freaking out about last? I think actually all of them, right? Real Fun we were freaking out about last time. Yeah, I'm pretty sure it was all of them because you show such an insane range. Like here, the wolf is so different, right? From like this piece here. Yeah, it's, yeah, absolutely incredible stuff, Real Fun. Like, mind-bogglingly different, but they're all really good. And so it's, yeah, it's gorgeous artwork. Is this y'all had a pop-up? Nice. That's awesome. Look at that. I love your logo too, by the way. That's super cute. Oh, there's the BB-8. Look at that. What a nice setup too y'all had. I wish y'all did it in New Jersey, then we can go see your setup. We have a local art festival too, and like a monthly little place that you can do. Get bored and jump. Listen, Real Fun. I 100% feel you. I think the being excited and moving around and is a real thing. It's like you just jump around. And you're excited, and then you jump to the next thing, and then you jump back, and it's all over the place. So for the Raiders coming on in, before I show you the science thing, we were talking about a new species of leaf bug that was identified, and then Ubi Iworks right there in the chat is gunning for a correct prediction today, and he made me bring out the guitar because he wanted to see the guitar, and he keeps saying he wants to be serenaded, but we're not going to have him serenaded because Smikes tells me I'm not tone deaf. My father tells me that I am tone deaf, which makes me very nervous to have any kind of audio sent to anybody of my own vocals. So, Music Girl, there is in fact a guitar here. It is in fact real. It needs to be tuned. One of the chords is totally off. But there it is. There is the guitar. I'm actually going to return this back outside because I just can't. Grimly, I don't believe you, Grimly. Grimly, I would say you are able to sing like a beautiful acoustic Johnny Cash. Is what I envision Grimly you're singing as. Beautiful voice, Grimly. I will not dare take any more of it. Guys, I'm going to put this guitar back, and then we're going to continue, and we're going to round out this leaf bug, and then we're going to get into possibly solving aging. Yeah. I know Smikes, but my father was the one that's like, hey, who sings that song? And I'd be like, oh, Godsmack. He's like, great, let's keep it that way. Right, that's my dad in a nutshell about the auditory cues. That's why, Smikes, you know, I have this part in me as to like you're tone deaf or something like that. I don't know. Let me take the guitar back and then we're gonna round this out. Smarty Personas. Thank you for the follow. Welcome into the colony. Well, according to him, Smikes, he was also very good at music and he was also really good at painting. No! Real fun. You smack them across the face. No. And I don't mean, like, actually physically harmful. I mean with, like, I don't actually know. I'm not advocating violence. I just mean, don't you dare have them say that. That's very rude. They should say, you know, they should say, real fun, who sings that? And then you could be like, oh, Godsmack. And they'd be like, no. You do. Because you've made it better. And I am blessed to hear you sing that song each and every day of my life and to be with you. That's what they should be saying. They should not be telling you to knock it off. Absolutely not. Real fun. Real fun. I will talk to them. Bring your significant other on. We'll have a chat. You sing louder? Good. Good, real fun. I am proud of that answer. I was not able to do that to my father. Although one time he walked in on my sister and I singing a Kesha song. My sister and I loved singing along to Kesha, if you remember Kesha, because she had very fun songs. And my dad walks in and he sees us like singing along to Kesha, and he just comes in and he's like, I thought I raised you two better, and just turns around and leaves. And I guess he thought that we were just gonna stop, and instead we just went right back to singing the song. We're like, well, he's already disappointed. It's fine. Let's continue. That's what we did. Guys, I'm going to take this back. Ubi, this has been a special edition for you, good sir. All your wishes? No, they haven't, Ubi. Ubi, you keep insisting that we need a company Tesla. Once I get my hands on it, when we make it in this business, then we'll get you in the company Tesla. And then at least another one of your wishes will have been true and come true. All right, so for those coming on in, we were talking about 7 brand new species of leaf insect discovered, 4 of which in India, 3 of which in the other parts of the Southeast Asia. Through genetic sequencing and morphology. Genetic sequencing primarily, there were actually big differences in morphology, or not big differences in morphology that made it actually difficult to tell the differences. And then we were looking at a video that was indicating differences in the size of these animals and how they sway in the wind to blend in with their surroundings.
From the same tree, living its whole life.
And so actually you can see in this stage here, that is the nymph. So that is earlier on in development, and then here is a larger one, a full size, and actually they undergo, they shed their exoskeleton, and that's a type of molting that occurs in this insect. And as they molt, you can actually see these patterns on their body that like looks like damaged leaves. They actually have a higher percentage of that every time they molt. I think is really remarkable. Yeah, Gwen, 7 new species that were discovered. And Gwen, what was interesting was, again, the crux of this is there's 7 new species, but it had been thought that in the whole of India, there was maybe 1-2 species max. And they actually now discovered 4. And the reason that was important is because the conservation biologists were saying, well, we don't need to really worry about conservation right now because there's these 2 massive populations and everything's fine. They did the sequencing and they're like, no, there's actually 4 species, and it actually turns out that some of them are now threatened and near being endangered because of how they're like a habitat loss. And so it's redefined conservation biology in the country. And if you take into account, and it's because they look very similar, and you can't necessarily differentiate them based on morphology alone, but the genetic sequencing has them quite different. So if you look at the phylogenetic tree, which is like Think of that as like a family tree of life that they were comparing the genetic sequences on. Here it is. Highlighted in green are the new species they discovered. And you can see they're pretty spread out along the tree. They're not just all clustered together. And so it's really neat to be able to have these species of like significant enough divergence to be able to identify them as, in fact, true branches of this phylogenetic tree. Just found out identifying guitars is more difficult than identifying insects in amber. Guess would be a Ivan's Art 300 Black Cayman. Thank you, Cliff. Cliff, I have no idea of the model number anymore. It's the only guitar that I have. Which, Cliff, there was a hot second where I started going to this guitar. What is that guitar store called? I think it went out of business, right? I don't remember. A guitar something. And I was like very tempted to start getting different axes. Guitar Center, thank you, Ubi. And Lita then moved up to New Hampshire, and then I didn't go back to Guitar Center for a long time, and then we lost the business. You might unfollow. No, Luminology. Luminology. Honest to God, I need to tune it. I need to see. We probably need a new amp because of the flood. It probably nerfed that amp. I do not know where actually the connecting wire is to actually hook up the guitar to the amp. It's somewhere in the disaster that is still our house because the basement still isn't fixed. Luminology, we need the disclaimers because I am very scared and have terrible performance anxiety. So I'm coming up with every excuse to not have to, because yeah. The cable, thank you, Ubi. It's been that it's been that long that I've even talked to someone about this, that I do this, because I'm that like petrified of showing it for people. Okay, so let's finish out this video. It's the best I know. No, Luminology, I know very well what I said. I know very well what I said. Guys, there is the nymph, there's the adult.
Like a leaf. Its brown spots getting bigger. It may never move from the same tree, living its whole life as a cunningly concealed copycat. Hey there, it's Lauren.
I do love their movement. So again, that's the movement of the nymph going from the ground up to the tree. And it's blending in, like trying to blend in like a swaying leaf. I have a second video that we can watch. It's not much more informative than what we just saw, but it's just a very short little look at this guy has a handful of leaf insects. And I just think they're pretty. I don't know which species. They're from Malaysia. We don't have a species of them, but again, it's really cool. And what's interesting too is, Ubi, based off the paper, there's a suggestion that there might be more than one species in this handful. I'd say probably not, given that it might be some guy's personal like pet collection. But there is based on the paper, there might be something like that. Yeah. If he throws his hands up, does that make it? It might, Luminology. It might actually. A bouquet of leaf bugs. Yeah, it would be. It's very pretty, right? They're just beautiful insects too, and so it's really cool that there's another 7 that have been identified. All right, so that is paper number 1 of the night. Paper number 2, I want us to get to, because I am very, very excited about this paper. It doesn't make a lot of sense yet, but let's take a look at it. We're all getting old, right? I bet they never leave him alone. Optic nerve. Well done, Optic nerve. Well done. Yeah, the scale was definitely helpful, Ubi. Absolutely. So, this is our second paper of the night, and it is from the journal Nature Aging. So, Nature is one of the top tier journals in the world to publish in. So, you have Nature, Cell, and SciAnts. Nature has subsidiaries. For example, Nature Biochemistry, Nature Aging is another one, and these are pretty still high-level publications, high-level journals. Real Fun needs to bounce. Real Fun, thank you again for the raid, sharing your community with us. I would love to see the new pieces once you're ready to share them. We'd love to see them. Again, have a great evening. Appreciate the heck out of you. Gwen, how dare? There's no bad jokes. Gwendolyn P, what bad joke have we had tonight, Gwen? Where it was like actually a bad joke, where it didn't make you just crack an ever so slight smile, and a little, maybe even you even sat back a little bit with your friend Chelsea. You had a proverbial chuckle with one another. You looked at each other. You're like, there they go again. Shoot, these dang scientists. And in that sense, the moment that you got that little smile, it's already a good joke. There it goes. Thanks. Well said, sir. Been freaked out lately because I think we're called wolf spiders have invaded my home. Big ones, size of small tarantulas, when you can hear their feet run across the floor. That is pretty scary. Could they kill me? Are they dangerous? I do not believe so. Do not quote me on that. But my knowledge is that no, wolf spiders cannot kill you. But don't. I will fact check that, Luminology. Shoot me I know you shot me a DM of your address. Shoot me a DM of your question as well, so I can do some lookup on it, and make sure that I will double check and see like what level is that there are, if that's okay. Because I don't want to lead you astray. As Derping said, I believe that there are some aggression differences between the wolf spiders, but I do not believe that there is anything like, physically damaging. Hi, Nerduino. Welcome in, Nerduino. Guys, if you like science, then go follow our NASA Space Scientist Extraordinaire, the legendary Nerduino. Has a Mini Weedo, has a Mrs. Weedo, has everything, in fact. Go follow Nerduino. Breathtaking Soul. We had a panel at TwitchCon about the science on Twitch. Nerduino, welcome in. We had a massive derail. Smike says that my taste in music is hilarious. Not in a good way. We even brought out the guitar. There's a whole thing. And we got through our first science news story, which is a new species, 7 new species of leaf insect. Now, this is our first scientific one that's beyond a new species, and I thought this was a very, very exciting study. So there was a paper that came out many years ago, and let's play the video, actually, of this paper that summarizes.
In dreams, one is not
Debs!
by earthly limitations.
What does that mean?
Come.
Debs, sleep well. Sleep well, Debs. I hope the metal gets better. And oh yeah, that's why we got in a musical derail, Nerduino. Debs said that she liked heavy metal, copper, and then we just went down that rabbit hole hardcore. So really, it's all Debs, Nerduino. The lovely Debs Bretto. Debs, sleep well, and I hope you get some good rest and you feel better soon. Debs super loves metal. That's very true. NASA is in the heezy. Gwen, did you know NASA now has a special sound alert? Hi, Epoch!
Jebediah!
Jebediah! That's apparently a sound alert now for the lovely Nerduino. No, exactly, Nerduino. Listen, I am but new at this. I do what I can, and I can what I do. Epoch, welcome in. Epoch, we're just about to start on topic number 2, which is a very promising anti-aging study, and it is essentially platelets a component of your blood, human blood, in wound healing, has been shown to cross the blood-brain barrier and induce neurogenesis, or growth of neurons, in the hippocampus, which is the learning and memory center of your brain. And it's been even narrowed down to a particular platelet gene and product of that gene, where scientists injected this into mice and essentially de-aged their brain. Now, I wanted to take 10 steps back and tell y'all where this science began. Before I do that, I want to shout out the lovely Epoch. Glorious gamer, artist, handsome as ferret. Go follow our friend Epoch. Epoch, a few years ago, there was a study that was about transfusion of blood from young individuals to old individuals. And Epoch, you might be like, Blent, this sounds very much like the Middle Ages, when there were, questionable queens in middle Europe that were harvesting young virgin females and drinking their blood and bathing in their blood. And you might be like, this sounds very similar. And in fact, there are jokes about this, that it sounds very similar to that. But the weird thing was that it actually worked. So we're going to watch this video, and it's, again, giving old mice blood components of young mice. Now, this video is a little bit newer, so we're actually getting to some of the components of what's being transferred. But let's just take a look at it and see.
You might be amazed to think about the power of blood. If you look into anecdotal stories all over the world, everybody knows blood is important, for our youth or for our health. And so it's almost like, I'm always joking that our discovery may reveal the secret of vampires in maybe Twilight stories or...
Smikes, did you see this? Smikes is like, oh, certainly this can't be Twilight, and Nerdowino, certainly this is Dracula. Sure enough, both of y'all are right, and there is both Dracula and Twilight mentioned in the same line in the same video. Gotta rewind it a little bit because I got excited.
It's almost like, I'm always joking that our discovery may reveal the secret of vampires in maybe Twilight stories or something.
Vampire.
No, I'm joking, but this is serious.
Serious because Dr. Shinichiro Amai found a shred of truth about blood as it relates to youth. His focus is on a protein circulating in our blood that declines as we age.
Called NAMPT, N-A-M-P-T.
Contained in small particles called extracellular vesicles, or EVs. Circulating in our blood, the extracellular NAMPT is linked to a key chemical regulating the aging process.
NAMPT is basically a very important enzyme in our body, which produces a very important chemical for our body functions. It's called an NAD, nicotinamide adenine dinucleotide.
And as NAD declines with age, so does energy metabolism, causing age-associated problems. Amai's Aging and Longevity Lab at Washington University School of Medicine in St. Louis conducted studies. By taking extracellular NAMPT, called eNAMPT, from the blood of younger mice and injecting it into older mice, something exciting happened.
Those EV-injected mice move around very actively. And then on the other hand, the control old mice are kind of like, slow and just like old people. So there is a stunning difference between these two individuals.
The enzyme from the blood of young mice extended the lifespan of older mice, and the benefit can be clearly seen when looking at this.
Nerdowino this guy seems very energetic for his age. Nerdowino, he might have children.
One mouse has a shinier fur coat.
So yeah, this image is very easy to see the differences. You can tell this one's the more ragged, older mouse, and this one's looks like it's the younger mouse, but in this image and this test, these two mice are the same age. It's just that one had the transfusion and one didn't.
And is healthier looking, whereas the other looks shabby and older. They're actually both the same age, 29 months old, which is the equivalent of late 80s in humans.
When we maintain the ENMPT levels in mice, we see a variety of anti-aging effects in several tissues. We're able to maintain the level of free running activity almost 12 months younger. 1 year younger means a lot for mice. If I dare to extrapolate to humans, it could mean like 70, 80 years old in people could maintain like a 40, 50 years old level of physical activity.
So the extrapolation there, right, is difficult to really nail down because he's trying to suggest that from a particular, like the 80-year-old might function like a 40 to 50-year-old. That's a very, like, it's a hard estimate because we do the same thing in fruit flies as well. Like a 60-day-old fruit fly is the equivalent of a 60 to 70-year-old person, but it's not always totally synced up. So it's an estimate, but it could work in that sense. So Noki, actually, we'll get to the follow-up, but this lab actually collaborated with another lab, and they found 2 additional components in the blood that are also adding and helping with life in different ways. So this original study, so what they did was they did the blood transfusion at the very beginning, Noki. And at that point it was just, does this do anything? And they found that yes, older mice look like they're younger on many different levels. In this study that he's talking about, they're actually getting to a factor. They're actually getting to a gene product that is demonstrating that, yes, if we have this gene product, they're able to increase their activity. He had mentioned nothing about their neural circuitry or brain potential, right? It was just much more of a physical aging, not necessarily cellular aging. So that's what the follow-up study is going to be. So it looks like you're going to need a combination therapy to really stay young in many different ways, if that makes sense. One-time deal, or do we need a constant supply of the product? So Nerduino, they did a couple of different approaches in the original paper. They did a one-off injection, and then they did also subsequent injections, and there was a significant difference between one-off and consistent, but both had a heightened level of, or an extended lifespan. So at least a one-off treatment is sufficient to induce a change. It seems like it's really timing based that needs to be identified. Once they figured out the cofactor that was involved, they just went to injecting that factor, right, instead of injecting the blood. So that's what you're trying to get down to, is that you're trying to identify, like, there is this factor here in the blood. What is that factor that I can then turn back age on these? Care about the extended lifespan, but if basically before until I die. Yes, Nerduino, yeah. If the mouse goes to Burning Man and packs extra MREs just in case. Ubi, I do not understand that reference. I know Burning Man's some kind of event. That is the extent of my knowledge. But remember, I'm not very hip like you are.
Something like that.
MI says continued research is showing even more promising results, which is expected to be published in 2022.
Our discovery about circulating ENMPT for its amazing effect of anti-aging, that means something very interesting and important, of course. So that's why, now I believe that blood has a very interesting power for our health.
So Gwen has a great question. Let me just pull up the mice image because I think it's very striking. So Gwen, the current paper identified platelets. And when they originally did the transfusions, it was total blood transfusion. It wasn't just platelets. The question was, Gwen, when they did the total blood transfusion, the factors that they identified in this paper here, that molecule that he was mentioning, actually cannot cross the blood-brain barrier. And so that's what inspired this next paper. This is the paper that came out, Gwendolyn P. August 16th. Right here. Always wondered this. Is there a method to seeing the differences between testing in humans and mice will be? Shark, no. It is... You're testing in mice because it's going to be... Or rats because it's closer to humans than if you test in a fruit fly. The reason you test in a fruit fly first is because it's a lot cheaper to do it. And if you see something positive there, you can move into... you can move into the higher order mammal like a mouse or a rat. After you do that and you see if there's an effect or a positive effect, you move to humans and you do injection treatment of healthy humans first, and to see if there's any off-target effects, and then you move into sick people. So there's like that line of way of doing it, but like, it's not predictable in how it's going to work. Sometimes it might work perfectly in a mouse, and you put it in a human and nothing happens. You can do in human cell lines, but...
Together, we can rule the galaxy.
Little Oxide, thank you for the resub! How you doing? Welcome the heck in! Hope all is going well. We're talking about this new study that came out, hopefully helping with aging. And fixing aging is the goal. Nerduino says, so presumably an older person... getting a transfusion from a younger donor is not that uncommon, so isn't there a way to already track possible effects? Ah, Nerduino, you get a very good, interesting question, sir. Concentration. Concentration is the issue. So, when they did the original experiments, we're talking near total blood transfusion, where they replaced near all of the blood within a old mouse with the blood of a new mouse, meaning Nerduino, the younger mouse was sacrificed for that, right? And so it was just a full-on difference. And actually, Nerduino, we tried to replicate it in fruit flies. When I was a grad student, we thought it'd be fun to do a replication experiment, but the issue was transfusing a fruit fly is really difficult when you are not in a very wealthy lab. And again, the reason for that is we were using like micro syringes, but they need to be pressurized with CO2 tanks in order to have any chance of sucking up the hemolymph for insect blood from one and transferring it the other. We did get a transfers to work, like hemolytic transfusions, but it just wasn't enough to, like, they were injured after that and they died from injury, and it just didn't work well. So it's the concentration question that you end up having to do. And so what's nice, though, is in these molecules, if you identify the molecule, you can get the concentration of the molecule right, and you can just inject that. And it can be toned to body size and shape, and there's those extra components that needs to be done. And presumably there's other components that needs to be done to, like, age and maybe even neural state. Like, maybe you can dose it on that level. It's still early enough because, like, it just came out to ask, and we're not entirely sure yet on that front. Luminology, I'm trying to find pics of the spider in my house. Isn't even close. Stumbled upon a picture of a spiny-backed orb weaver. I've seen orb weavers. I'm not sure if I've seen that one before, though. Spiders look like they went to the garage and got... Oh, very cool, Luminology. That's lovely. Thank you, Nerduino. Nerduino, a very, very good question, because you're right, like, if it presume it exists, but apparently it's just not enough blood. And I guess the other thing, too, is if there would have been, like, very massive transfusions... Human studies are notoriously difficult to do because you can't control for things. Right? Like, if you're trying to do that on a human, diet, exercise, lifestyle, genetic background, all of these factors come into play, and you have no idea what's influencing it. So if it works or doesn't work, is it because there's something else that's happening, or is it actually what you're predicting? There's just too many variables happening. So you could do, like, what tends to happen in these, like, if we did have the data in Nerduino and it was the right concentration, you'd have to do what's called a meta-analysis. So you'd have to reverse track back and see, okay, when did this patient get the blood, and then how were they, what measurements did we have when they were administered before the transfusion, what measurements have been taken after the transfusion, and, you know, because they didn't just look at lifespan in the study, they looked at motor function. And so that's the data piece that you would need to have as well, and it's just going to get really murky really quickly. But once there is human testing, it's still, as Epoch says, it's really murky because there's going to be, you know, dietary differences, lifestyle differences, again, the genetic background differences, but you'll start off with a much more controlled set of experiments and monitoring and making sure that there's either no off-target effects or you know what they are. And yeah, it can happen. It's just, it gets messy really quickly. So I really liked this story for a lot of different reasons. Kind of feel like someone's out there, grandma got run over by a bus and now he's running marathons. You know, but if we found one person like that in Nerduino, would it be because they got run over by a bus, or because there's other factors at play and we wouldn't know, right? With an N of 1, yeah, exactly, you're right, they could just be what, the who, I'm watching, I'm not gonna get it. I didn't get it. Constantine works though too, because he brings back people from the dead. Alright, maybe. Can we choose the person to experiment on? So Ubi, presumably, like, so I would say these are still a little ways away from going to human trials, but if there are signups, they try to find, again, phase 1 is usually college students, to be totally honest with you. It's like really young, really healthy people, and they just want to dose you really high and see if anything bad happens. And then phase 2 is a more select group, where you might have some that are ailed and see, like, if you can help them or not. And then phase 3 is a little bit more opened up. But yeah, I don't know, Ubi. I don't know if we'd be able to get any of us in there. Because, see, it's okay because they gave you $200. That's very true. It's very, very true. Yeah, no, Luminology, that is the first time that guitar has ever been on stream. That is the first time that guitar has ever been on stream in over 2 years of streaming. It has never been bought on stream before until tonight. And I've been told by the mods they're deleting the VOD. Yeah, that's very true. Yeah, Shark. Sleep well, Epoch. Epoch, thank you for being here. Galera Dragon, thank you for the resub at 8 months. Thank you, Galera Dragon. I appreciate the heck out of you. How are you doing, Galera? Guys, go check out, again, the lovely and amazing, breathtaking Galera Dragon, the maker and crafter extraordinaire. I know no one said that, Smikes. I was being silly. I saw it in the background before on camp. It was on a picture, Cliff. It's never been in this room. Ever, Cliff Alistair McLean. Galera, how the heck are you doing? Thank you for that resub. Galera, we're talking about this potential treatment for aging. So this is the big paper that came out, and I was really excited about it. There were actually a series of papers that came out this past week on the topic. Not bad. I made my own zucchini. Oh, nice. Galera, zucchini bread or banana bread? Which is your favorite of those two? I asked them clustered together because Lita, when she makes those kinds of bread, at least when she did in New Hampshire, she'd make like a banana bread and then she'd make a zucchini bread as well. And her recipes are great because they're like, it's like really moist and flavorful. And at first I was like, no, zucchini bread's going to be terrible. It's really, really good. I still think I lean more towards the banana bread, but they're both really tasty. And again, I never thought that zucchini bread would be tasty, but it is really, really good. I never had like a different one. Oh, Luminology. Yes. Balint cooks a lot. Balint barbecues. Balint does Hungarian food and more as well. I'm not much of a baker, but I do cook. Probably banana bread. Yeah, pecans and walnuts. Yes, absolutely, Galera. Slice of each with some Irish butter. What is Irish butter? Nor do we know. Want to see a cooking stream? That's fine, Luminology. I need more equipment to do that. I need more equipment to do that. We do not have the setup at the current stage to do it. And actually, Luminology, Lita and I had talked about, because when Lita was on every Friday, we used to have a cocktail of the week that she would do for Science News Friday. And we had been talking about it'd be really cool to cut to camera in the kitchen that have her on screen where she does the cooking, or like the mixing of the drink and talking about the science behind it, and then we can cut back. Is one thing that we were thinking about. Yeah, I mean, Luminology, the current sub goal right now is to get us to unlock a new emote slot. And if we do that, we will raffle off this plushie Kevin. This one right here will get raffled off. If we hit 10,000 followers, whichever one comes first. So that is for one follower goal. At the end of this month, Luminology, we're going to do a mini subathon. We tried playing the video game Grounded, which I really wanted us to play to talk about the science behind the game, because there are a lot of insects and it's a great opportunity to learn. Our computer cannot handle it. So we need to, if we're going to do it, we need to get an Xbox Series S, which I know sounds like a hilarious issue to have, but if that's something we want to happen, then that's what we want to try to do. So we wanted microscopy muffin Monday. Well, if Lita makes a muffin, then yes, then we can't do that. Galera says bake is made of zucchini, onion, garlic, cheeses, breadcrumbs. Ooh, that sounds amazing, Galera. And absolutely, Madame, always. Okay, so to get y'all excited, this study, there were 3 different techniques that were looking at Hi, Giraffe Killer! Guys, the legend, the man, the myth, the most wholesome, handsome, and kind man on Twitch has graced us with his presence. He is the lovely, the amazing Giraffe Killer, a.k.a. Gafafe. Please go follow Giraffe Killer. His name actually stems from... it stems from the fact that he went to Africa during his teenage years, and because he was very passionate about being a biologist, and especially conservation biology, and he learned about that giraffes were actually endangered because of poachers, and he went to Africa to protect giraffes, and he actually took out poachers, and he's called Giraffe Killer because he dressed up as a giraffe, so poachers wouldn't see him, and he wiped out a bunch of poachers. He also is an amazing musician, Giraffe Killer is. He made the hit single It's absolutely incredible. Now you're gonna say he went to Africa. No. Derpington, he took care of the giraffes. The poachers are trying to shoot giraffes. So Nerduino, listen, that's how committed this gentleman is. He used himself as bait to catch the poachers, Nerduino. That's what Giraffe Killer did. Can you imagine how dedicated you have to be to conservation of a whole species to put yourself in the firing line of poachers? Man's an absolute myth. A legend, in fact. And again, he's a great musician. He's a great friend. And I love his face beyond all. Seriously, y'all, go follow Giraffe Killer. He's an absolute hoot. He's a great guy. And he is kind, wholesome, and handsome beyond all reason. Please go follow him. No. So Nerduino, he does not have a long neck. He's a regular human-sized neck. He actually, the suit, his face was at the base of the neck, like there was a peepee hole that he can look through. And on top of his head, it had the rest of the the giraffe like neck. So yeah, he was able to like use little binoculars down below, and then if he wanted to, he had like Nerdowino, like on a submarine, how you have the periscope. He had that come out of the giraffe's eyes all the way at the top, so that he can look around as well and like get like a really long distance vision. And then when he saw the poachers, he actually burst out of the suit and took out the poachers. So actually, that's why the giraffes still talk about the lore of giraffe. Like, he is still known by the giraffes in Africa, and that's why the giraffes are no longer endangered. They're only threatened. Presumably the torpedoes. No. So Nerdowino, torpedoes are illegal to obtain in Africa. You actually have to be a pirate to be able to do that. That's like that Tom Hanks movie. So instead what ended up happening was Giraffe Killer used soil as well as thorny bushes to take them out. Yes, shark arresting. Yes, and by arresting, he took them to St. Peter. Yes, to the pearly gates where they were judged. Giraffe Killer is not. Well, he became judge and executioner at a certain time, but he's not the official full-on judge. That is St. Peter. And so they went to St. Peter, and I don't know what happened from there. Neither does Giraffe Killer. That's not our business. That's between them and God of killing people and killing animals. It's a whole thing. And yes, exactly. By execution, we mean arrest. Exactly, Nerdowino. And after that, we don't know what happened. Chow, exactly. Hey, Pete! How's it going? I'm glad y'all worked it out. I'm glad. I'm very glad. See, yes. So, again, please go follow this man. He saved giraffes. Also, he's a great musician and a great friend of ours. One way or another, I'm pretty sure I'm never meeting Pete, because you're not going to die because of the scientific paper. That's an excellent point, Nerdowino. Thank you for getting us right back on track. Great job, Nerdowino. Thank you, Little Ox, after the eat. So, Giraffe Killer, this is a really cool study.
We'll all be Gucci.
And it is a way to slow down or reverse aging in the brain. And so the way this started was they were doing blood transfusions on young mice to old mice. And they find that this blood transfusion to the old mice made the old mice slow down in aging, if not reverse, to the point where they looked like young mice and they behaved as young mice. Now, they were then trying to figure out, well, what are the components of young blood that make these animals reverse in aging? They found in an earlier study that there were things attached to metabolism. But when they injected those gene products into the animal, it didn't hit their brain. Like, it didn't fix their brain. Their brain still aged, so they still got neurodegenerative disorders. But physically they changed. So, like, they still had the activity of young mice. So then the follow-up study was, okay, how can we fix cognitive disorders? Like, what is the factor in young blood that is there? I think you have to remember about your stream. Thank you for the Lord Kubi. Pretty much any word or phrase can have a totally different meaning no matter how obvious it sounds. Is that a good thing, Luminology? Also, we're very welcoming here, and so that's why if you're talking about Giraffe Killer's name, yes, he is very kind, wholesome, and handsome. So, Giraffe Killer, there were 3 studies, and they were all trying to identify what this factor was. Now, the factor that came out, this is the wild part, it is called platelet factor 4, PF4. No, so listen, Nerdowino, so I heard an example of why we go to the fridge. Nerdowino, is going to blow your mind. Y'all know how I don't know about y'all do, I do it all the time. Do you go to the refrigerator, open it up, and then just close it back and you don't end up getting anything? I heard a comedian say that opening up the refrigerator is the save point in life. That's when you go save your game, is when you open up a refrigerator and don't get anything. That's like the game of life. And that's why we will randomly go to the refrigerator and open up and get nothing, because you're just saving your progress. And and I still do it. I did it today. in fact. And I'm like, dang it. Better Self tells me it's that I'm bored rather than I'm actually... That's fair, Andrew, but I do like that it's a safe spot. I also like to imagine that it's probably because I'm bored or that I'm hungry and there's nothing to eat. And there is, but it's just like nothing I want. It's a whole thing. I do not. Even, I can't remember what I opened it for. I always take something out, even if I don't need it, people might be watching. Do you have, Nerdowino, one of those fancy, like, LED refrigerators that have, like, wireless and, like, system that you can play Pandora off of? There's, like, is the refrigerator that tells you, like, what you're out of? Like, that measures, like, with some kind of AI of what's inside and, like, sends to your phone, like, you need to go pick up some milk because you're near out? In that case, I would absolutely get stuff out every time, because I don't need my refrigerator knowing that. Sumo Torpedo is something from Steven Seagal movie, but nope, it's also an electric ray. Oh, that's fair, Luminology, that's fair. Play Minecraft in his fridge. Yep, that's fair, Galera, too. Do not, because I just need a fridge to keep stuff cool. I do not need to text my fridge. Nerdowino, what's cooler than texting your fridge? Oh, got him! Opening up the fridge because I'm bored is such an adorable human mannerism. Like, I know, right? Truckhorn. Can you play Doom on... Oh, God, Derp. I love Doom. No, knock him out. We got him! Got him! Alright. So, Draft Killer, you mentioned... You asked, like, what the heck? It is a platelet factor that is helping your brain. Right? Cliff says, I have a very energy-efficient refrigerator. Means it's really small on the inside because of the insulation. It means there are always things falling out if I open it. So then, Cliff, you're always doing something when you open up your refrigerator. Like, you're cleaning stuff up, right? So at least it's better than me, where I honestly will open it and will look, and I'm like, well, there's nothing in here that I want. Yes, so the platelet Draft Killer, as you suspected, it's an immune cell. And it's used in wound healing and clotting. So it's a type of, again, like if you cut your hand or your skin anywhere, how dare? God, no. Y'all, there's someone naughty here.
Ah! You are wholesome as ferret. Yay!
Go follow Mr. Sean Solo Beats, musical genius extraordinaire. Your resident gross boy, as he calls himself. He's very, no, listen, Draft Killer, Mr. Beats says things like, he calls me the most handsome Draft Killer, Mr. Beats does, and he lies, because, in fact, he is, in fact, the most, now he's being naughty, that needs to be wrecked, because then he sets lies in the stream, and it's terrible. I don't like people lying in the stream. It's a whole thing. So please, don't be naughty, Mr. Beats. I love you, Mr. Beats. You are, and yes, you are extremely handsome. Sean, I hope everything's going well, my good sir. Sean, by the way, Sean has a Discord, and in the Discord, one of our friends, V tries to chat. There is a channel in Sean's Discord called V's Sleeping Corner, where I regularly see our friend V in that voice channel, and it has his mic off, and I presume he's just sleeping in there. I really want to log in and scream on that channel. Like, I just want to have, like, a big scream and see, like, if there's any reaction from V. Because I think that would be really funny, but I'm like, I don't want to actually wake the guy if he's really sleeping, but can you imagine? Or I guess just start whispering sweet nothings. Either one would be a fun one to do. But yeah, Sean, I saw him doing it today. I saw him today in the channel. I was like, man, I really just want to go in there and say hello. But I'm like, I don't know. I don't know if I should. Anywho. Sean, this study is looking for a way to reverse aging in the brain. And so they identified this factor called PF4 in platelets. And so it's an immune cell Draft Killer, but platelets, so white blood cells will actually go and respond to an already existing immune challenge, where if you have they recognize the antibodies and have antibody production, so it's part of the adaptive immune response where they're looking for a pathogen, whether it be a bacteria or a virus that has come before and has infected in the previous times. There's also natural killer cells or neutrophils, and those are your phagocytes. So those are what eat up the original site of infection. That is your innate immune response. They eat up or phagocytose the stressor. Platelets are not involved in either of those. The platelets are involved in wound healing and clotting. So if you cut yourself outside, the reason you stop bleeding is because you form a clot. You clot up that wound, right? And so platelets are the cells that aggregate at a wound site, and they'll heal you up, essentially. So people with hemophilia, that's a clotting disorder because the platelets aren't functioning properly. And those have been, if you are at all familiar with the Russian oligarchical families past history, they tended to all have hemophilia. That's the time of Rasputin. He was taking care of the royal family that had were hemophiliacs. One of the original fancy places. Yep. Okay. So exactly. So Giraffe Killer. The question is, what is the connection between a cell type and a gene product that's involved in wound healing and rejuvenating your brain? Right? And so it was really cool. Actually, I wrote down some quotes from an interview. And so the lead author goes, this is a quote from the lead author. He says, "My first reaction was, what do platelets have to do with cognitive advancement? This is crazy." The lead author is like, this makes no sense. And that, to me, is a really, really cool start to a study. Because if you get a result that doesn't make sense, that means you replicated it, so it's not just an accident. That means there's some really cool science at play, and you've stumbled upon some really cool biology. Gray, you can. Guys, Grayscale is an amazing artist extraordinaire. Please go follow the lovely and amazing Gray if you aren't already. Love your face, Gray. Even though I've never seen your face. Only seen your hands. You have wonderful hands, Gray. So, Gray, the goal here is that they've done this. So they were doing transfusions from young mice to old mice. And then eventually they figure out there is this component of that blood that they're transfusing that's acting on the brain. And what's, again, wild is it's a clotting factor. What's interesting about it too, Gray, is that this clotting factor, unlike other mechanisms that they tested, can cross the blood-brain barrier. So a lot of molecules, we were just watching a video where, again, they identified a different component in the blood that helped the animals, when injected into old animals, they behaved and moved and looked like young animals. Meaning, Gray, let me actually show you the video image. So, Gray, can you tell by eye the different, which one's the old mouse and which one's the young mouse? So let's say this one's mouse 1 and the other one is mouse 2. Can y'all guess and just tell me left? So this one, mouse 1. Only because who's left, right? This one? Yes. Okay, so I assume we're talking about this one. It does look older, right? But these are actually the same age. Except this one got injected with that blood component, and this one did not, and the far one did. But they're the exact same age. And so not only does their physical appearance differences, and they're 28 weeks old, which is the equivalent, 28 months, not 28 weeks, 28 months old. That's the equivalent of an 80-year-old person. 80 if you're scaling up to human age. And the only difference is one was injected with this compound that they found that was helpful in blood, and the other one wasn't. The other thing they noticed, in addition to physical like appearance changes, hi Mogul Hawk, welcome in. In addition to physical differences, their activity was also different. So the ones that got injected with that compound moved like if they were young, like they ran around as much as they did. So Gray, the genetic background is, I think, is what you're asking about. Just to make, tell me if I'm not understanding correctly. When you have these mice, you order them from a company, and they're all genetically identical. And so when you test them, like let's say this is B56 mouse, they're all genetically identical. They're highly inbred. So if you get a result like this, it is due to whatever you've injected and not due to the genetics in them, because they're genetically identical. Now you might be like, Blint, if they're inbred, certainly there's things that are wrong with them to begin with, right? Because inbreeding is usually bad. There are inbreeding issues. For example, Gray, my favorite one was there was a learning and memory study done on mice, and the learning and memory study was done by playing a musical note and pairing it with electric shock. And so then eventually the mouse learns that this sound means their pain is about to happen, so they get scared and they freeze if they hear the sound, right? And so the researchers were like, we found this gene that if you get rid of, the mice are no longer, they don't remember and they're not afraid. So like this is like the master regulator of learning in a mouse. And it turned out, Gray, it's because they were deaf. That was the inbreeding effect. The mice were deaf. And so it was not at all due to the genetics, like, of what they thought, but it was due to some inbreeding effect to it. So now there's actually a lot of controls, as you said, Giraffe Killer, you have to do for these experiments. So Giraffe Killer, you're 100% right on many different levels. You have to do a sample size that increase. Now, in mice, 5 usually is a good number. If you were to do this in fruit flies, you do hundreds. And that's in part because of money. So keeping around a mouse, aging a mouse, is about $70 a day to feed them, clean their cages, give them fresh water. $70 a day. And so, Gray, if we do 5 mice control, 5 mice experimental, and we age them to 28 months, that adds up very quickly. Humans are cheaper. I mean, oh yeah, it's like it's a lot. It's definitely a lot. So Galera, they're not necessarily clones. They still mate, but they are, and there's still some mutation that happens, right? But they're essentially genetically identical. You're gonna have some differences because if they were true clones, what you would do is you'd take the DNA out of a cell and implant it into an egg every time, and you'd be taking the same DNA every single time from an original host, implanting it into the embryo, because then it's like there's no chance of there being any difference. But here it's like sisters and brothers are mating, so it's like just highly inbred. You don't introduce any genetic variability, so it's actually closer than a twin study, Gray. They're called super siblings, which is about 75% identical, and that's the same amount of genetic identicalness that you would get like from a bee or an ant colony, and that's just because there's one queen laying and there's all these workers. Hi, Kathy! Thank you for the Kathy! So Galera, the reason it costs that much is because of the facility. So it has to be in a standardized setting. Light, dark cycles, humidity. It's usually the same person or 2 people taking care of them. So it's the animal facility handler's salary. It's usually their full-time job. There is also an animal facility that has a bunch of costs to make sure everything's standardized. So it's it's huge levels of buildup and cost that built in that adds up to be about that much a day. So it's not just that, you know, you put them in a box in the corner of your house and you give them some, you know, I don't know, wood chips and food and water, but it's like everything has to be standardized, clean. Like there's just so, and also they genotype them, meaning that they take like a small tail clipping and you do sequencing on them before you do the experiments. All of that's just like adding up, right? Yep, minimal changes in variables. And Gray, weirdly enough, maybe not weirdly enough, I don't know. You can tell me if you don't think this is weird. You can order these mice from a place called Jackson Lab. That's what everyone does, because you're like, I want to do an experiment on a black 56 mouse. That's what was in this paper. I go to Jackson Lab and I order B56 mouse. I get it. Great. What's wild is, Gray, to me, is that their diet also affects things. So the diet at the facility might be different than the diet at the lab that you have. And that could have an effect on your experiment. And actually that was discovered a couple, like 10 years ago, where the microbiome was different because of the different diet. And even though, because Gray, in papers, it says mice were fed standard diet. Fruit flies were fed standard fruit fly diet. But no one defines what that is. And so if your recipe is at all a little bit different, then you might have very different results. And it's like, ah! So that's, again, another big part of why it's going to be costing so much. So Jackson, yes. So the Jackson Lab, Gray, we can actually go to the website if you want. Okay. Google. Yeah, Jackson Laboratory. Thank you, Boga. So, yeah, Jack, right, great. So here we go, great. There's the website. You can search for mouse by strain, stock, gene, allele, synonym. So, and here's so you can do, here are the featured strains. So it's like, here's your black 56. NSG, the tau is a protein that mutated causes Alzheimer's. It causes platelets in the brain. And so that's one that's like heavily featured here. There's this K, and these are not mutations, right? The K18HACE2 is a particular mutational site. So, Gray, the idea is you can, in theory, order any gene mutated in the mouse, if it's not lethal. There are some genes, if you mutate in the animal, it just dies, right? So then you might have to make a new mouse where you turn it off in a time-controlled manner. So you allow it to develop, and then in the adult stage, you turn it off. But let's say you wanted B56. This is your wild type here. These are the kind of yes. And sometimes you might have to make your own. Might have to make your own on it. You're on your own if you want to be killing those draft killer. That's why I didn't go into mammal research, because I can't kill mammals. That's why I did mice. My dad was actually he worked in mice, rats, and opossums, and he had to kill them, and I was like, nope, I cannot do it. Gray. Draft killer's name comes from the fact that when he was a teenager, he went to Africa as a conservation student and protected giraffes, and so he's called draft killer because he took out the poachers that were trying to kill the giraffes. He is in fact a protectorate of giraffes. So please do not call draft killer as any kind of like hateful person. He is one of the best people in the world, serious, and he's really handsome. Shark says, so how do they do cancer tests with mice? So Shark, you can mutate a gene and give them cancer. It's that easy. You can also you can transplant a human cancer into a mouse and it will have a tumor. They tend not to naturally get cancer until they're old. So what you'll usually do is you might do a targeted mutation in a group of cells, and that's where you'd have the mutation form. Noki, do these mice retire, and can people adopt retired mice? So Noki, I don't know if you can adopt them. They do have what are called retired breeders. Hold on. We can actually maybe find it here. That might be a different tab. Where a live mouse. Aged mouse. Here we go. So Noki, so you can buy an 89-week-old mouse, one mouse, Noki, for $546. One mouse. Now, Noki, these are retired breeders. So at a certain age, they no longer breed the mice and they just keep them alive because people might want to do an aging experiment. And can you imagine, we just talked about the price, right? So imagine, Noki, I want to do an experiment on an aged mouse. It's important for me that I do this experiment, otherwise I don't know if it's worth doing the experiment. Is it worth me taking 3 years or more to age a mouse and test? What if I'm wrong? Right? And so you go onto this website and you can order them, test it, and see if you're right or not. So they're called retired breeders. Noki, there are laws in place about the welfare of these animals. So if you're worried about how they're kept, there are many laws in place on animal welfare. Density of the cage, for example. How often food is changed, how often water is changed, cleanliness of the cage. All these things are really at play. And so you'll never have... it is not allowed to have an animal suffer. And even when they're having to be put to sleep, it is done in the most humane way possible. This isn't just Jackson Lab, this is all U.S. institutes have these what are called IRB protocols. And each university has one where they have to abide by both the NIH guidelines, which is the National Institute of Health, as well as the university guidelines to make sure that the animals are not abused in any way. And that doesn't extend to every animal. It's called higher order mammals, so you have your rats, your mice, your birds. No for any insect that I'm aware of. No for bacteria. No for cell lines. Monkeys will have it. So things like that have these guidelines, but insects do not. Yeah, and so then here's your website where you can order them from. So let's say for the experiment you wanted to have an older mice. Yeah, it's pretty expensive. But remember Draft Killer, how much each mouse costs a day to upkeep. Really, they're not making almost any of a profit here. Moving on to slowing testing, is anyone developing testing methodologies that are more similar to humans without having to rely on other mammals such as rats? There is what is called the organoid system. And the organoid system is essentially you're growing an organ in a dish, but it's not a full organ because it doesn't have blood vessels connecting to other things. It is just a cluster of cells that have been differentiated to be a heart or a liver or something like that. And that is something that folks are working on and trying. It just seems like there's a lot of complications with that. That there isn't enough things done correctly. There's too many, because it's not in the animal. Your organoid dish, like your fake organ in a dish, can give you some answers, but it might not be the most informative. It could be very different when you go to the animal. But so we're still dependent on the animals, but there are methodologies that are being worked on that work in a different way, which is quite cool, I think. So to get back to the paper, I was telling you all this, what I thought was a cool quote. Yeah, Shark, give some idea before you move into the animal component. So from this lab, the last author, the Dubal, they were looking for a hormone called Clotho, and they found that was linked to longevity, and it boosted brain function. But they found, so Draft Killer, this is where we get to the platelets. This Clotho molecule could not cross the blood-brain barrier. So it could not get into the brains of the animals to make them younger. And that's where this platelet came in. So it turned out that what they thought was actually happening was when you injected the mouse with this Clotho gene product, this protein, the platelet factors increased. So Draft Killer, think about it like a some kind of like connection web where you increase Clotho, that leads to an increase of this platelet, and that leads to better cognition. And so they found, like, when they analyzed the brains, they found that it's this platelet that crosses the blood-brain barrier. So that's the catch, right? Of like when you find a potential target that might be working, they have been studying Clotho, right? Because they inject Clotho and the animal gets younger. But it turns out that wasn't actually for the molecule. It was a downstream, like, next step that was the true molecule that was doing this. Andrew, thank you for the lurk, you handsome devil. I love your face. And so I think that's quite remarkable that they were able to identify it, but also, like, you're finding out the pathway, right, of this gene X on this gene X on this gene X on this gene, because then you actually know the mechanism of how it's working. Because to me, Draft Killer, there's always, with, like, new drugs that are discovered, if you ask your doctor, how does it work? This isn't them being Raiders! Oh my goodness. Professor Astro Canuck, Raiders!
SciAnts stream, but we ain't bugging. We over here buckling. You be chugging while we
Hi, Astro.
Tribes arrive. I promise that I have prescribed you vibes. The vibes we got are giant. Shout out to the stream for the raid. That's SciAnts.
Raiders! Welcome on in, Professor Astro Canuck. Astro, how the heck was your stream, my good sir? I mean, I was lurking on you, as I always do. Folks, if you're not checking out Astro Canuck, you're not doing life right.
The galaxy.
Dame Karen, thank you for the resub for 20 months. Thank you, Dame Karen. Guys, Professor Canuck is a...
we can rule the galaxy.
Oh, Astro Canuck resub too. Oh my goodness, it's a double resub. Astro for 16 months. Dame Karen for 20. Thank y'all for the resubs. Welcome on in. Guys, Professor Canuck, if you don't know, Lego builder extraordinaire, amazing astronomer. If you want to look at the night sky, if you want to learn about the planets, the star systems, he's got some amazing telescopes, takes beautiful images, and will teach you all about space. He's also a graphic artist, does amazing artwork, custom Legos, you name it. He's also a published author. If y'all don't already, there is a link right here in this command to a book by Astro called Beetle Blues. And he did all the illustration work. It is beautiful.
The galaxy.
Mogul! Thank you for the resub with Prime for 2 months. Mogul, thank you so very much. Y'all kicked off a choo-choo as well. My goodness gracious, y'all kicked off a choo-choo. Fun night talking about supernovae and how they evolve over time. Watched a Falcon 9 launch. Nice. How was the launch? Was it successful? Let me welcome everyone in as well. We have Dame Karen. Thank you as all for the resub. Hello, Coolie Spots. Welcome in. Orange TJ, thank you for the 100 biddies. That means we're going to have a raffle in a moment. Uncle Jimbo, hello. Karen Day, hello, hello. Scorpion Gucci. Goddess Toya, welcome in, first time chatter. How you doing today? Guys, and Fire Florida 225MF, welcome on in. Good to have you here, first time chatterer. Successful indeed. Nice, Astro, excellent. Pretty Astro Canuck is in fact very rad. Gray, thank you for the biddies. Guys, I'm going to play a quick channel trailer, tell you about who we are and what we do here. We also do raffles for every level of a hype train completed. While the trailer is going, I will set up the raffle, and we will have some raffles for stickers, and I'll catch you up on what we're chatting about tonight. Don't go anywhere. We'll be right on back.
Welcome to SciAnts Streams. If you are interested in all things science in a fun, wholesome environment, you are in the right place. SciAnts is a unique husband and wife team of Belinda and Lita, who hold their PhDs in molecular and systems biology from Dartmouth. What can you find here? The SciAnts team is dedicated to making science more accessible to our entire community. They believe that it is the responsibility of scientists to engage with the general public, and Twitch is one of their media doing this. By showcasing their own research, highlighting new discoveries, answering viewer questions, and sharing their passion with you, we do a variety of interactive science streams, including microscopy of ancient organic insects, 3D printing, art, science news and deep dives, the occasional game, and more. If you've ever wanted to have a fun space to nerd out, now is your chance. We're streaming daily science content here on Twitch. understanding not just what we know, but how and why we know it. Check out the About page for more information. And consider hitting the follow button to join our colony and our mission in science for all.
Oh my goodness, and a double rate as well from Astronomy Show.
Science Stream,
Hi, y'all.
but we ain't bugging. We over here bubbling. You be checking what we got. Now the
Hi, Astronomy Show, Raiders!
Promise that I have prescribed you vibes. The vibes we got are giant. Shout out to the stream for the raid. That's science.
Raiders! Welcome into the double raid Astronomy Show. Welcome. How was your stream? Hello, Misadventures in Astro. Welcome on in. Guys, we just got rated by Astro Canuck. Please go follow Professor Canuck. And now we're going to get a fancy pants shout out to the lovely Astronomy Show as well. We are also looking at the night sky. Tell me, Astronomy Show, of all the things that you were looking at tonight. Again, for the Raiders, my name is Blintz. Tonight we're doing Science News of the Week. And we're in the midst of a hype train. If y'all would like to win a sticker and or a magnet, type exclamation point raffle. If you win, we will ship them out to you. It is international as well. For every level of a hype train we complete, we do more raffles for stickers and or magnets. Complete a level 5 and I put on a ferret onesie, and we do raffles for hard enamel pins and sticker sheets that look like this. And if we complete level 10, we do a custom one-of-a-kind SciAnts mug for you as well. And a piece of 30 million-year-old amber with insects. Never any pressure to get us there, folks, but those are the things on the menu. Raiders coming with Professor Canuck.
Hello there.
Hi, Grayscale. Ray, you've been here.
Hello there.
For those coming with Professor Canuck, tell me what was your favorite part of the stream tonight. And same coming in with the lovely Astronomy Show.
Barbecue sauce.
No, you're totally Gucci, Gray. You're totally Gucci. Yes, Ferret onesie, orange TJ. If we complete a level 5, then these things pop up the menu as well. Misadventures in Astro. Thank you for the tier 1 gift sub to Trixmot. Thank you so much for that gift sub, Misadventures. Trix, I hope you enjoy the ad-free viewing and the emotes made with love. For the Raiders coming on in, tonight we've been talking about the cool science news of the week. We started off with 4 new, count them 4, new species.
It's all Gucci.
Of leaf insects. And now we're talking about a brand new genetic study that was identifying factors that will help you age slower. In particular, neuronal features, so slowing down cognitive disorders. And it seems to be very promising. It's working really well in mice. We'll go into the science behind it in just a moment. As soon as we finish our hype train, we do some raffle. We'll get back into the science of it. Astro Canuck, Astronomy Show, thank you all so very much for the raids, for sharing your communities with us. Folks, I cannot endorse these two people highly enough. Both Astronomy Show and Astro Canuck are educators. Astro had some really cool images of Jupiter that he posted on his Twitter as well recently. With Astro, I love that picture. I also love that you were doing it was just a great shot, and of you as well, my goodness. Orange TJ. Oh my goodness, saving the hype train. Saving the hype train with 5 tier 1 gift subs. And Astro, can I pull up the image, by the way? Would you mind? To Tripwire Gaming, Gemini CXC, The Latest Kate, Adam Wiley, Fishman 001, Orange TJ. Thank you for those 5 tier 1 gift subs. And Astronomy Show said we covered all things astronomy, star parties, telescopes, new science papers, and space exploration. Very cool. That's awesome, Astronomy Show. Guys, that means we're going to have at least 2 raffles. Let me quickly find Astro Canuck's Twitter. I want to share all this picture that Astro took. And again, if y'all have any images you want to share tonight, anything is Gucci. Hi, Tripwire.
Hello there.
Astro, I know you're on the other platforms as well, but I still find Twitter to be the easiest. This one. So this one was an image that Astro had taken last night.
This is true.
And this just turned out beautiful. Like this is like dream stuff, y'all. Orange TJ, thank you for another 2 tier 1 gift subs to Sketty Sandwich and Banzai Merlin. Orange, thank you so very much. That unlocks another sticker raffle, so we'll have 3 sticker raffles at least that we'll be doing. And again, if we complete more, we'll do more raffles. Yeah, Noki, I love this picture. First of all, Astro looks extremely handsome, and I get to meet Astro Canuck at TwitchCon. I'm so excited. We're having a science meetup on Saturday, I believe, at TwitchCon, and Professor Canuck has said that I can take a picture with him. Professor Canuck has said that. And guys, Astro was one of the very first streamers I started watching on the platform. He was the second science streamer I ever saw. It was Paleontologizing and then Astro Canuck. And a staple of my weekend is Astro Canuck's Lazy Lego Sunday. So it usually starts at 10 a.m. Eastern on Sundays. One of my favorite streams of his because it's just relaxing, great to put up. And then I love all the science content. And just hanging out with him is always a positive sign. It's always a good time. And this is one of the pictures, again, I was absolutely adoring, was this one here with Astro showing off Jupiter. Gray, let me show you something, Gray. Gray, there's a secret you might not know about. There's a secret, Gray, you might not know about, Gray. How dare, Astro? I'm not too kind. I am but speaking the truth. There is a Twitch panel called The Scientific Method Twitch Edition. And Twitch has selected these people to be part of it. So there is Moo Hoodles, Nerduino. Our host is a streamer by the name of Nicotine. Paleontologizing, us at SciAnts Streams, and Volcano Doc are the panelists, and Nicotine is our moderator. And so it'll be on 3:30 p.m. Pacific Standard Time on Sunday, and we will be live streaming it from this channel. Yes, Derping, I will give you the link. We will be live streaming it off our channel, so y'all will be there in some way. Y'all will be there in some way. Dame Karen, this is the moderator that we have, is Nicotine. They are a gaming streamer. We met with them last week. I think we met with them last week. And we have to submit slides and imagery and all this kind of jazz that we'll be chatting about. The whole mission is to get people excited about science. Yeah. Pattern, I had no input on who the panel should be. We submitted an idea for the panel, and then it really became like it was a weird selection process. So it's not about, deserving or not. It's they ended up picking. What I really like about it too is that it's not all partnered streamers. So for example, Nerduino and Volcano Doc are not yet partner. And I'm hoping that this will catapult them into that level as well, because having more educators on Twitch be partner is a huge step. And so I think it's a nice mix of astronomy, paleontology. We have the biology, we have volcanology. I think it's going to be a good time. It's on Sunday, Gray. So it's on Sunday, 22nd at 3:30 Pacific. And Pattern, there's going to be a science meetup as well. At that Saturday, the day before, it's like a meetup networking event for everyone who wants to be like science and technology. So I think there's gonna be cool folks there as well. I don't know who all is coming, but hopefully I'll see like Professor Canuck there and other science enthusiasts, and we'll, Astro said he'll take a picture with me. We'll see if he wants to. Actually, Astro, maybe I can bring my copy of Beetle Blues. Astro, can I bring my copy of Beetle Blues and have you sign it for me? I've been hounding Astro to get a signed copy of his book, but now I'm like, oh, I can just take his book and ask him to sign in front of everyone. I'm like, sign your book, please. Guys, 20 seconds left on the hype train. ! Raffle to get in on the raffle. And we'll pull some winnings. An hour. What do you mean, Dame? Is that like a good thing or a bad thing? Dame Karen, I've read that book to my my daughter. Okay, that's how much I enjoy that book. Look, Gray. I fan if I meet anyone. My fellow streamers on this platform, I love promoting and shouting out and being excited about my fellow streamers. Because if there's any opportunity to promote y'all, and if I can make a small difference in your journey to what you want out of this, I would love to do it because I believe in y'all. Right? So, like, I don't know, it's just important to me. And, you know, my dream is to make this full-time one day. I imagine there's others in the community that's their same dream. If I can help with a shout-out and, like, show off your stuff and get other people excited about you as well, then heck with it. Of course I'm going to do it. Guys, thank you all for that. Level 4 hype train. Appreciate all the crazy support we're getting tonight. Y'all, let's go ahead and do some raffles. Let's go ahead and do some raffles, folks. It is open international, so if you are not in the U.S., you're totally fine. You don't have to pay a thing. I just need your mailing address. That's all I need. And unfortunately, I do need your mailing address because it doesn't. I have the merch here. I have to package it up myself. Another streamer friend going. I cannot wait to see. Are you going, Gray? By the way. All right, let's go ahead and pull the first raffle. In 3, in 2, in 1. Dame Karen! Dame Karen, congratulations. Congratulations, Dame Karen. Dame Karen, which one would you like, Dame Karen? Orange Tea, you're going to be at TwitchCon? Heck yeah. Please come up to me, Orange Tea. Dame Karen, what would you like, madame? 1 through 13 are stickers. 14 through 17 are magnets. What would you like, Dame Karen? And Dame Karen, I will need your mailing address, please. It has been a little while since you won something, and I want to make sure it's going to the right place. Yes, Luminology, we have 2 more raffles. No, you're fine. Luminology, we got 2 more raffles. We got 2 more to pull. 2 more to pull. What would you like, Dame? 17 magnet? You got it. The amber magnet. Awesome. Dame Karen, could you either DM me on Discord or Twitch Whisper? And we'll get this shipped out to you. I will try tomorrow, but if not, we'll do it early next week to be able to get it out for y'all. All right. Next up, in 3, in 2, in 1. A truck horn. A truck horn! What would you like, a truck horn? Find you and request consent for a hug. Orange, that sounds perfect. That sounds perfect. And thank you for asking for a hug. I love hugs, but I just want to... It's always better to ask. Even though I've asked Astro, for example, like 30 times now, and I'm going to ask him when I see him as well, because I'm like, well, what if he changes his mind? I don't ever want to get it to anyone's face. 17, you got a truck horn. I think you've sent me your address recently, but would you... I'm so sorry. If it's not too much, could you resend it? It helps me keep track of things. All right, y'all, let's do one more. We got one more raffle to do. Thank you, a truck horn. One more raffle to do. In 3, in 2, in 1. Mogul Hawk. Mogul Hawk. 1911. I love it. Yes, Mogul Hawk, you want a sticker or a magnet. What would you like, Mogul Hawk? Mogul Hawk, what would you like? So, again, 1 through 13 are stickers. 14, 15, 16, 17 are magnets. You get to choose whatever you want. Except the golden sticker. If you want the golden sticker, we got to spin the wheel for it. And then you get whatever the wheel lands on. And that's not... I don't control the wheel. The wheel knows all and sees all. 11, you got it. Purple Kevin. Mogul, would you mind sending me your mailing address?
Where's my drink? What drink?
And we'll get this shipped out to you. Just send me that mailing address, and I'll get it shipped out to you. Guys, I'm feeling zesty. Can we do one more tonight? Can we do one more tonight? Would it be okay to do one more tonight, folks? Would y'all be alright with one more? Maybe. Possibly. Potentially. Let's do one more. Can we? Yes, Derpington. Let's do one more. Let's do one more. In 3, in 2, in 1. Misadventure in Astro. Misadventures in Astro. Congratulations, Misadventures. You have won a sticker and or a magnet. Misadventures, what would you like? And I will also need your mailing address to be able to ship this out to you, but you get to pick whatever you would like. off the menu. Number 3, purple Twitch words. You got it. Excellent. Guys, thank y'all again for the hype train. And yeah, Misadventures, if you can just shoot me that mailing address, please. Question, y'all. I have a question from Lita. Is this cute for my daughter? The first Halloween? Or this one? I kind of like the skeleton one better. Which one? The pumpkin or the skeleton? Okay, I'm seeing a lot. I think it's the skeleton, yeah. Lita said they're on sale today, so she was wanting to get a little something for them. Coolly, I'll show you again. Both are good. They're on sale today, but I asked her, she grows so fast, she'll be grown out of it by the time she's, you know, like 3 weeks later. There's the pumpkin. And then there is the skeleton. Yeah, Noki, honestly, it's just a size thing. She grows so fast that it'll probably be like a few weeks and she'll be out of it. And baby clothes can be pricey. Madness of how pricey some of it is. So, for sure, skeleton, awesome. That's what I told Lita. Alrighty, so, y'all, again, thank you for that hype train. And make sure, folks who won, please send me your mailing address. Again, that's Misadventures in Astro, Mogulhawk 1911, a truck horn, and Dame Karen from that last batch. And then we've also had Shark, Marianne, Luminology, Shipwreck tonight. And I've gotten most something from everybody so far. Oh, thank you, Noki. I think she's pretty cute too. So what we were chatting about tonight, y'all, is a really exciting paper on longevity aging factors, where having this particular... So the original, to bring everyone back up to speed, there was a study a few years ago where they did a transfusion of young blood from a mouse to an old mouse, and it made the old mouse young on a physiological level. And so since that, they've been trying to figure out what factors are in young blood that make the old mouse younger, right? And so they identified a couple of factors. One was much more for physical and locomotion. So Dame Karen, you can actually see in this image. Dame, can you tell me which one is the old mouse? Look on this image. So let's say the one closest to me is mouse 1. The one further away from me is mouse 2. Which one's the old mouse, Dame Karen? Or anyone in chat, you can tell me. Mouse 1 or mouse 2 is the one further away? 1 probably. Yes, Dame Karen. So this one looks older, right? Its hair is thinner. It's thinned out. It looks a little scruffy. This one right here. Turns out, Dame Karen, that they're the same age. These two mice are the same age. That one up there got some of that transfused blood from a young mouse. They actually gave it a particular factor. That was in the blood. They identified what the factor is, and it made it so they looked younger, and they also acted younger. Their locomotor behavior was much more in tune with a young mouse than old. These two mice are 28 months old. That is the equivalent of an 80-year-old person. And so it works. And so Dame Karen, since then, and other folks, anyone who's interested, of course, Karen Day is very excited for it. By the way, folks, if you're not checking out Karen Day is my name, you're also not doing life right. Pyrographer, artist extraordinaire, and singer. Go check out Karen Day is my name. What's really cool about this is they've started to figure out what the components are in the blood that are important. And in this particular study that we're chatting about tonight, it's a really neat story because it started out as there was this gene that they identified and have been injecting into the mouse called Clotho, and it made a protein product, and it made the mice young. However, they did not find Clotho in the brain. What that means is that that molecule is not crossing the blood-brain barrier. But what they find though is the brain that gets these injections looks younger. They are not finding cognitive breakdown, like neurological breakdown, like of the neurons and the structures in these brains that have been treated with this compound. And it's a natural protein product, so it's synthesized, yes, in the lab, but the genes for it are turned on in the young mice. Does this have anything to do with the study on turning back the hair loss? No, not at all on this one, Dame Karen. Not at all. Nell, thank you so very much. Go get some good rest. Good luck on the Green Comet, and have a great day tomorrow. Appreciate you being here, Nell. Thank you so much. And you too, Gray. Sleep well. Thank you all for hanging with us. You are the most competent blood-brain barrier. Oh, thank you, Dame Karen. I missed the word sayer the first time I looked at that comment. I was like, it almost felt like, it's like, well, you're a blood-brain barrier. Because, sometimes it's funny to be like that with people. And, I was like, oh, I don't know where Dame Karen's going with this, but let's go along for the ride. So what they find is, thank you, Cooley. Appreciate you. The hippocampus of the mouse brain. Hi, Lady Lara Croft. Welcome in. No sorries, Lady Lara Croft. I will not nerf you for that, because you also said hi. No sorries, Lady Lara Croft. Welcome in, ma'am. So they found that the hippocampus of these mouse brains, which is the learning and memory center of a mammalian brain, not just a mouse. Oh my God, I'm so sorry, Lady Lara Croft. I did not do that. Your comment was nerfed by a mouse. It wasn't me. It was not me, Lady Lara Croft. But listen, we do have rules here, and I guess the rules have to be followed by just every. I'm sorry, Lady Lara Croft. I don't make the rules. No, not banned, TJ. Lady Lara Croft said I'm sorry for being late, and honestly, if you spend you are on whatever time you arrive is the time you were meant to be arrived. Spending any time. Wow, the nerf didn't work at me. Oh, sorry, Dame Karen. Sorry, Karen. It's because you added someone. I think I don't have the command so that you can add at anyone. I think that's why I didn't trigger. You should be able to trigger it, though. You are subscribed. No, so TJ, I just don't like that folks say sorry, because there's no pressure to be here. Whenever you come in is the time you're supposed to be here, and there's no reason to say sorry, because I am blessed that you have spent a moment of your day with me. Right? There are a bajillion different things you could be doing, and a bajillion other channels on Twitch that you could be at, and you've chosen to spend a moment of your time here. That is something that I cannot even thank you enough for. Right? Sorry for being on time. Or you're always on time. Okay, so this hippocampus, right? They found evidence of neurogenesis in the hippocampus that from individuals that were treated with this compound. What does that mean? Neurogenesis means neuronal growth. So there were new neuronal growths of those neuronal cells in the hippocampus of the brain of these animals that were treated. Meaning their brains looked like young brains. Now, again, what we were really excited about before y'all Raiders came in was that they had also identified earlier within another study that there was a clotting factor called PF4. It's on a platelet. And a clotting factor is something that's wound healing. So if you have your hand cut, eventually you'll clot and you'll stop bleeding unless you have hemophilia. You stop bleeding, and there apparently is a factor there called PF4. They found this PF4 in the brains of these individuals, and it turns out that this cloth gene Clotho, carries PF4, and it carries the PF4 to the brain, to the blood-brain barrier. The Clotho can't cross, but the PF4 can. And so that's the key gene. So it's really cool because they thought they found the gene, because if you were injecting Clotho into the mouse, it was reversing its aging in the brain and the rest of the body. But it turned out the way it got to the brain was it had to carry the PF4 to the side of the brain, the blood-brain barrier, and then it crossed over. Pezzy, you're not wrong. Clotho is also linked to other disease states, which is why you have to be careful whenever doing this kind of dosage. And it's always a question of how something like this works. But it's very, very promising. Gwen says, how do you say I'm sorry in pretty Magyar? Elnézést. Professor Canuck, sleep well. Thank you again for the raid. Guys, please go follow Astro Canuck. Lego builder extraordinaire, graphic artist, published author, astronomer, get your guide of the night sky. Follow our friend Astro Canuck. Thank you again for the raid, my good sir. Love your face. I'll see you real soon. Like, Sunday, at least. If not tomorrow, I guess. Are you streaming tomorrow, Astro? Astro streams a lot like we do. And so I just have notifications on and I'm always there. He will, look, Karen, if I'm lucky, he will. He may moon you. He doesn't like to moon me. I don't know. I think it's because I'm married. Which is fine, because he respects my marriage, and I appreciate that. I really do. I really do. So we did have the question of how long have they been doing this? Karen Day asked. So, Karen, the original study, which was just blood transfusion, and it was from young mouse to old mouse. That was the first paper, and that was circa 2016 is when the paper came out. I can't tell you how much before that they were working on it. Probably a couple of years. If I had to bet, I'd say this is probably going on a decade total. Especially because mouse research tends to move slowly, because aging them and keeping them is a lot of money and a lot of time. And there's since then have been many other papers that have come out. So the very first one was just, here's blood from one to another. It has a positive impact. There's the first paper. And proving that is paper one. The next papers are figuring out what are the molecules in the young blood that are different from the old blood, and what do they do. It was a theory. I think, Karen, I don't remember the interview where I can find the interview with those original scientists that did the transfusion, but in modern interviews, they joke that they are proving vampires right, because vampires suck blood, or was it there were queens as well and other royalty in Europe that also drank the blood of young women to keep themselves, or bathed in it, right? And so it's all these stories from my historical past. It's well, they weren't true in that context, but it seems like there are factors in the young youthful blood that is, the issue is in old blood and older individuals, there's this thing called haploinsufficiency, where if you have two normal copies of a gene, if you mutate one copy and you're young, you will not see an effect, because that one copy is enough to transcribe and make protein that you're totally fine. But as you age, you start to be less and less efficient when you're transcribing your genes and making proteins. And so if you're haploinsufficient, meaning that you have one mutated copy, you might start to see that one mutated copy and the one regular copy not be enough as you get older, and then it just goes downhill further and further. And so that might be an effect of what we're seeing, is that you have high levels of it in your youth, but then it gets turned down during old age, and that's what leading to the aging process. And so I think it was a theory that they figured it out, but it is a quite wild of a theory, right? But it's pretty cool. Could this help with dementia? Yeah, so Lady Lara Croft, their goal here is all kinds of unique neurological disorders. And they do also suggest, and this is kind of crazy, but why not? Because if this PH4 factor induces neuronal growth, they suggest, and they haven't done any experiments on this yet, but if you have a severed nerve cord, right, and you can't walk, imagine if you can induce neuronal growth to reconnect those neurons. And again, they haven't said that that's something that they can do, because none of the experiments have been on that. But in the discussion of the paper, they mentioned that could be a possibility. Again, given how this works, because you have the neuronal growth that starts in the hippocampus, and to keep it young, maybe there's a way to co-opt the system to induce neural growth to reconnect, severed spinal cords or other injuries along those lines. That is at this point total speculation, but why not, right? Like, why not test it? I think they're still going on a lot of mechanism on this particular study of like just looking at just the brain. Fix the shoulder. My shoulder lacks cartilage, and that's coming from bone marrow. And apparently, that's why my last 2 surgeries, they drilled into my shoulders with like a diamond drill to have the bone marrow ooze out to try to re-put in cartilage, and it failed. So, but maybe, like, there might be another mechanism of the blood that's perfect for mine. Because one cool thing is that they've shown is that in these animals, other factors in the young blood do different things. So not everything's neuronal. Some of it is locomotion. Other is, like, we saw this mouse a minute ago, right? I asked y'all which is the old mouse and which is the young mouse, and it turns out they're the same age. It's just the scruffed one didn't get an injection and the other one did. And so, that might be something where musculature can be restored as well. And, maybe instead of doing like the microfracture surgery with the drills, maybe that's something that can be restored as well. So, like how, and you start getting vibes of Fountain of Youth, right? And like rejuvenation medicine. A big issue is always knowing how to turn these things off. So cell growth for rejuvenation is great. Uncontrolled cell growth is cancer, right? So it's always that balancing act of dosing, concentrations, how to get that to be on point. Now, the other coolest thing that they found in this study is that actually there were, so there had been a previous study from this that we're looking at. So, Karen, I'm giving you a little bit more of the history like you asked. Is again, they did blood, it was the very beginning was the blood plasma, or they did the straight blood between individuals. Then they also did blood plasma. And plasma, they found, rejuvenated the brains of elderly mice. And then this is that connection where they found that Klotho gene carries a blood plasma component through the blood-brain barrier to have some regenerative capability to it. What was cool was the way this paper came about. There's 2 professors on this paper, and they actually co-published the papers simultaneously. And you can see there's actually another one here, which I'll pull up in a second. But Klotho, a longevity factor, improves cognitive function in aging non-human primates. So this Klotho gene that we're talking about here in mice, I'll pull up the paper in a second. They did the same thing in monkeys, and they found the exact same thing, right? So then if you're, maybe skeptical of like, well, it's a mouse, a monkey's even more closely related to a human, and it also had very beneficial cognitive effects. They did not test and make sure yet, at least in that study, if it's also that clotting factor, if it's the same clotting factor that's actually being transported to the blood-brain barrier and then across, but presumably it's going to be the same one. Little scary. Dame Karen, my guess is it would work. The dosage is going to be the big issue, though. But it's these 2 researchers. They came together and they actually they were at a conference, and so in the interview they were like, we had independent studies and converged on this PF4 the way they were doing it. And they were talking they were just having coffee, and then they kept talking. So one of them is a neuroscientist at the University of Queensland, which Dame Karen knows about because it's in Australia. The first team found that if mice exercise, they have an increase of PF4 in their brain as well. So they actually found evidence that exercise keeps the mouse young, which is kind of wild, right? And it's through the same PF4 as they found is the transfusion, right? So there's a way to artificially increase your PF4, at least in mice, by exercising. And it also goes into the brain of these animals and keeps and actually showed new nerve cell growth. And the other lab was like, well, we did the same thing except we injected PF4. And they're like, okay, y'all, we got to chat. And they were like, we got to publish these papers together. And so they found a link between the immune system and aging and cognitive decline. Yeah, so Orange TJ, at least hitting the gym until we get this in an injectionable method. Which I think, again, super, super cool, something like this says. Their quote, and I think this is a good quote to have, is that PS4 is a promising new player, but it's only one part of the complex puzzle. And they say, these discoveries greatly enhance our understanding by unraveling one factor. We always want to know the full puzzle, but understanding each factor fills in a piece. And it's a very important thing to note, because there are other factors at play here, right? If the original experiment was, you're just transferring blood from one individual to another, and it reversed aging, there's probably going to be a lot of stuff there. Work out 5 times a week. There you go. See, Karen, you're Gucci. Gucci, Karen Day. Other factors that were at play that I looked up that have also been identified. There's a gene called GDF11, and that has been linked to calorie restriction. So if you and it varies because it's mouse, right? So it's hard to actually like say how much it would be in human. But restricting your caloric intake increases this gene product GDF11, and that also seems to have some restorative effects, not in the brain, but in like that physical appearance, which is an interesting one as well. They say that the limitation of the new study, which I like that they mention, is how well it's going to eventually translate to save something in there. Free longevity factor paper. What is this astronomy show? Oh yes, actually, astronomy show. I had it here too. I already had it pulled up. So actually, what was neat was these were published in like back-to-back issues. You can see here the associated content between the two. So yeah, I had both ready to go. I will post the papers on the Discord, as we always do under the mention on stream category. So y'all, if y'all are interested, you can see and read the papers and the videos we watch as well. But what they found, and Karen, you'll like this, both our Karens, is that they did, in the study, they did measure P4 levels, or PF4 levels in humans, and they did find that PF4 levels decrease with age. Now, granted, their sampling of human, this is always the difficulty of human sampling, is that it wasn't a very diverse population. So it might be possible that populations from different parts of the world will have different patterns, but they did point out at least the population that they looked at had declining PFA. PFA is paraformaldehyde. PF4 as humans aged. So it again is another point that there could be a link there. Then, as Astronomy Show pointed out, the second paper came out. Realistically, when would we be able to see this treatment available? So Karen, this paper, this other paper that popped out, I can't remember if they have a picture of the monkey. They just have a cartoon. They might just have a cartoon figure in here. They did this on rhesus macaques, which are, so if you do something like this in like a monkey system, it, for better or worse, usually tells us that it's going to be closer to full development and coming out in a human. It's still going to be, I would say, at least a decade away. If they're starting phase 1 clinical trials now, they'll probably start as well. Think of something unrelated. Does this mean people that have issues with blood clotting age quicker? Meaning, Shark, that if they cannot clot? Not necessarily. Hemophilia also isn't just a mutation of PF4, but there's other mutations at place there that's actually causing the hemophilia. You might still have PF4. It's just a clotting factor. It's not the primary clotting factor. That doesn't mean that your hypothesis isn't correct. There may very well be an aging effect if you have any kind of clotting disorder, but that's not what having a PF4 mutated will not lead you to have hemophilia. But that's not to say, again, that your experimentation, like, your question can't be true. I just don't know if anyone's looked at that study. But at least it's not a straight shot, right? Like, if you have this mutated, you have hemophilia. It's not that clear of a although that would be, right, a really interesting study, Shark, to ask then, because you can be like, all right, what's the follow-up? But yeah, unfortunately, we don't have that factor yet. So then they moved to this paper. So this was in rhesus macaques, and what they did was, they're suggesting that brains are more similar to ours, and they were wanting to see if PF4, eventually, if that's the improving factor. They don't know if it's PF4. They know PF4 levels decrease in a monkey brain and a human brain with age. They also know that in a monkey brain, in a monkey, injection of Klotho helps cognition. And they also did not detect Klotho in the brain. You could be like, it's like the transitive property in math where, like, this equals this equals this, therefore this equals this. I would say it's not a logical leap to say that we would probably be on the path of saying that we've identified PF4 as being important in humans, but they haven't done that full experiment. They haven't done pointing out in a monkey that this is that clean connection point between these animals. So there's still more work to be done to ensure that it's the right factor that's being identified. And it's still, I think it's super, super cool. They say, and again, they had an interview where they say, we know exercise is great, but you can't do it necessarily when you're older and frail. And you also can't do caloric restriction if you're older and frail, which is another reason why it's like, well, why don't you just tell people to exercise? It's like, well, you can't necessarily do that. If your body is breaking down, caloric restriction is not advised for anyone in that age group because it's so hard to keep weight on. And my grandmother is having this issue now as well. Like, she's eating less because she's older, and the doctor's like, you need to eat more because you are wasting away. And it is going to have a bad effect. And so you don't want to do that. Yeah, Shark, I thought we had a possible rheumatoid arthritis diagnosis on me. It just turned out I had nerfed shoulders. But yeah, like, exercising is hard, right? And so you're not necessarily in a position to do that. Like, it might be you give you this regenerative dose, and then maybe you're able to do exercise and augment it. We don't know yet. So, Karen, they did say that the human trials, they hope to start testing soon. Again, my suspicion it'll be it's usually a 10-year project to bring a drug all the way, like, FDA approved to be tested in a human. They did note in the discussion of the paper that they need to figure out how the PF4 acts in the blood versus the brain, and how that relationship will need to be tested in this, like, rhesus macaque setting and the mouse to make sure that it should be part of any dosing in a human, right? Or if it should just be Klotho, because again, if we're modulating a clotting factor directly, that could have off-target effects. I mean, so could Klotho, but it seems like Klotho has a tight regulation, and so if you're changing the levels of that, it will also change the levels of PF4. but maybe to the point where it's not going to have, like, necessary clotting down, like, negative effects. There's nothing to suggest that there are these negative effects, but I like the fact that they're very open and honest about it in the discussion and highlight that they should be, that one should not be overexcited. One should be careful when interpreting this data. There could still be other factors at play that you have to account for. And once you've accounted for them, then we can get excited about it and test in humans and, you know, make sure that helps in humans. Hi, Kennedy. Lucky in that regard with my mom, who is 90. If anything, I'd say, Mom, pace yourself with the food. She's not heavy, but the exercise is impossible. See, Kennedy, my grandmother is like gardening. The woman cut down a tree a few years ago, right? She's in her 80s. So getting her to stop is difficult, but now it's like she's just not eating and not drinking enough. And it's like a big issue because she's always so dehydrated. And it takes her forever to try to get something to eat. But so, I mean, that's most of the aging study that I was going to talk to you about. I had some videos about this, but I feel like we've kind of hit all the important points without any videos beyond that mouse video. What do y'all think? I don't really think we need extra videos on this particular topic. I think I hope. Does this make sense, y'all? This story? Yeah, and Cooley, she unfortunately also has Alzheimer's, and so it's a whole thing. That we're Alzheimer's and/or dementia for my grandmother, so it's a very difficult time for her and the family. But you know, we do what we can. Does that make sense, y'all? For that, I know this was like a pretty hard-hitting scientific topic, but I think it's elegant enough genetically where it's like Clotho does this, and we figured out it didn't do this thing. Instead, it was acting on PF4A, which is what ended up doing the actual effect of the de-aging process. And again, they just, you know, they did cognitive tasks, which, you know, they did a Y maze test on the animals, and they tested their memory. They did genetic sequencing to figure out what genes are responsible. And so here, if y'all wanted to see how this even looks, this is called, so panel C is a log fold change graph, where they're plotting the fold change against of the sequencing experiment they did. And so here are all the genes that are in this quadrant are the ones that are significantly changed in a positive direction. So if you want significance, it has to be above this dotted line. Let's actually zoom in. This is just like a neat way of visualizing data. It has to be above this dotted line, and it has to flank either the left of this dotted line or the right of this dotted line. Everything in here is insignificant, and everything below is not significant. So you're left with 1, 2, 3, 4, 5, 6 genes. GAPDH is already one that you're not going to test because GAPDH is a chromatin factor. There's probably no reason to test for that. And then you test the other ones and you see which is most important. And this PF4 is the highest in terms of fold change out in the right direction. It's not the highest in terms of log, the Q value, but it's a very attractive target to look at. None of us are going to escape aging. We age every day. Yep, absolutely. But, Karen, we might be able to slow the process, or at least, like, what I would like, Karen, you know how it's like you retire when you're fragile and old? At least a lot of people do. You can't enjoy travel, in my mind, if you're that, like, fragile, right? Like, I want to retire, and if I'm going to travel, be able to travel and enjoy those final few years. And it's just a frustrating thing that it's like, and, you know, honestly, Karen, I'm probably never going to be able to retire. The way that the finances, you know, and retirement plans always look like they're shaking out, we're probably never going to be able to retire. This is a sad reality, right? But I'd still like to be able to enjoy life in terms of traveling, exploring, and just like really having like that. I don't want my last few years being like I'm in a bed and not being able to move, or I'm forgetting things, like because my memory is shot because of Alzheimer's, right? So hopefully, even if it doesn't extend our lifespan by 30 years, the last years that we have, our quality of life will go up. And so while we can't escape aging, maybe there's a way that we can improve how we live. Or I mean, right, even as Shark just said, until we become cybernetic, right? Is just to make that Simplex! Why, hello, Simplex. Raiders!
SciAnts stream, but we ain't bugging. We over here buffalo. You be chugging what we got. Now the tribes arrive. I promise that I have prescribed you vibes. The vibes we got are giant. Shout out to the stream for the raid. That's SciAnts.
Raiders! Welcome on in. And Simplex, how was your stream? How was Astroneer? Tell me, what is Astroneer? Is it an astronomy game? Perhaps. Simplex, I want to know all the things. Welcome on, and how was your stream? What was the best part of it? Did you have a good time? It's great to have y'all here, guys. Go check out our friend Simplex. Here's a fancy pants pinky out. Shout out for N Simplex. Where we go? Where's Simplex? There you go. Boom. Cozy space game. Nice. That's awesome. That sounds like a great time. I was my most recent cozy space game, Simplex, which may or may not be space, is Pikmin 4. There's a little bit of spaciness to it. It's more like insect behavior, but it is still cozy out the way. I'm glad that you had a nice time. I'm glad it was a good stream, it sounds like. And I'm glad that you got some good cozy vibes from it as well. Welcome the heck in, Raiders with Simplex. My name is Blint. I'm 1/3 of SciAnts streams. The other 2/3 are my lovely wife, Lita, and baby Alona. We're research scientists, and we do interactive science streams here on Twitch most days of the week. Microscopy art, sci-fi science, science news of the week, deep dive scientific topics, games, 3D printing, and IRL streams. Pretty much do a little bit of everything, but all in the context of science. Raiders, welcome the heck in. Started with Undertale, making progress. Nice, Simplex. I appreciate the switch to something cozy, because that's where I would have to be if we were playing Undertale. Raiders, and actually, Simplex, as always, if there's anything you want to share with us, Mikasa Sukasa, a fun clip, anything at all, you can drop a link into the chat, or you can, like, if you want to promote an Instagram page, Twitter, Facebook, whatever, drop it into the chat. We'll happily promote it to the party people, and, or the Discord. Anywhere is fine to promote. Please do. Please share with us anything and everything that gets you excited. We were just finishing up this topic about this new anti-aging treatment when Karen said it was really cool, which was, I'm excited that y'all enjoyed it. We're just wrapping up with looking at some of the raw data that I just thought was kind of neat to see. So like how they identified the gene was they just looked at this graph and they found PF4, which is the key gene of interest, is one of the differentially regulated genes, meaning that it's different when compared to control, and it is statistically significant. And they just use these dotted lines, which are not always arbitrarily set, but like a smidge arbitrarily set, to figure out where the cutoffs should be for these things. They did a ton of controls, so you might be like, Blent, you know, this seems like it's a very big result of an experiment. How do you decide when it's a good thing and when it's a bad thing? And it's just, you just have to be very tightly controlling everything, and they did have those. We do math streams on Tuesdays at 9 p.m. Eastern. Ooh, what kind of math? Simplex, what kind of math do you do? Been a 2-year college professor for 15 years? Heck yeah, Simplex, that's awesome. Simplex, are you by chance part of the Knowledge Fellowship? That is a group of educational streamers. Here is the list. It's not a stream team. It is just a group of educational streamers. If you're doing math content, I would apply. You have? Okay, cool. Gotcha. I don't know if you're part of the list, but I would definitely apply, because that would be a great thing to have you be part of. And again, more educational streamers on Twitch. I don't know if I've seen a math streamer before, so you got our follow. Also, I like the element of the games too. That's fun. It's a really good time. So yeah, we're just highlighting a last little batch of the cells, some of the experiments. Let me see. I'll go back to the article page. Again, nothing too wild. I just wanted to highlight some of the data points that I thought were kind of neat. So again, they have they inhibit platelet activation, so they actually inhibited platelets forming and they found it was the opposite result, where if you don't have the platelets, then there's no increase in cognitive function, which is quite neat. Few mostly ended their careers on Twitch or stream so infrequently. Ah, gotcha. Yep, that's totally fair, Simplex. It's a hard thing to keep going. I mean, we streamed, most days of the week, 5 to 6 days a week at least, and I have a full-time job as well, and a kid, little 7-month-old. Goal is to eventually one day make this full-time, like more so than it is now. We're still a little ways away from it, but by a little I mean a fair ways away from it, but hopefully one day we'll be able to get to that point. I find being a science communicator and educator one of the most important things that we can do if we're in science. So yeah, there was the platelet activation that we chatted about, crossing the blood-brain barrier. Mine will be in a few months. Gotcha. They have that info. Hey, it'll be good, Simplex. So they looked at, the brain, and they looked at like where it crossed over, and they actually looked at the hippocampus, and they noticed hippocampal growth. So essentially, all the things that we chatted about, they identified are in the figures, quantitated, and I like that they did the imagery as well, especially because they were focusing on the hippocampus, which is again that learning and memory center of the brain. They also just injected the PF4, which is this last figure, and PF4 alone enhances cognition in young mice and aging mice, which I thought was like the extra crux. So Karen, if you're still there, if you inject the PF4 in a young mouse, even that young mouse does better than its young counterpart. So there's, again, it's a balancing act, because if you do too much growth in any kind of cell type, it'll lead to cancer, because that's what cancer is, uncontrolled cell growth. But the fact that it also improves young animals shows that this is a real thing, and it's really, really cool. I'll be doing videos of researching for the whole time. Oh, gotcha. Nice, Simplex. It'll be best of luck, my friend. Every time I say blood, now I got nervous, Dame Karen. Blood-brain barrier. There we go. I was trying to be silly when saying it. The sillies didn't work. So chat, that is paper number 2 for tonight. I think that's probably a good stopping point for us. We've been going almost 5 hours. I normally wouldn't sit. Becoming my new. No sorries, Dame Karen. How dare. I normally wouldn't say that this is should be a stopping point. We'd do another one, but ever since we've had the little one, sleep is a limited commodity. And so I think this one would be a good stopping point if y'all don't mind. Again, I'd love to keep going, but I do need to get some sleep before little one. I'm sure will be waking up early in the morning, as she always does. Usually between 6, if we're really lucky, 8 is what she does. Thank you, Astronomy Show. Astronomy Show, thank you for that raid as well, my friend. Guys, we're gonna first of all, I have to fulfill the prediction. Let me go ahead and fulfill that. Oh, go ahead, Simplex. Drop it. We got through 2 science news stories of the night. So tonight, with 18,000 channel points on the line. 18,000 channel points. The correct answer is 2. We hit 2 science news topics tonight. Let's see. Who won? Noki of Azeroth and 2 others won a boatload of channel points tonight. Congratulations, y'all. Congratulations. Again, we talked about 2 spikes. We talked about 7 new species of leaf insect, and we talked about the biology of these leaf insects, what they're like, what would be the ideal experiments to really prove that it's camouflage, and much, much more. I'm not fully satisfied with the experiments on that front, but it's your standard new paper experiment where it's just, these are the new species. We're not going to tell you anything else because we sequenced it, we did morphology, and that's that. Which is cool. But it leaves me wanting more, as it were. Don't accept unsolicited advice, see the irony. Everything's a phase. Good, enjoy the ride. It won't last. Bad, don't worry, it won't last. I like that, Simplex. Well done. Noki, yes, Noki, you crushed it on the on the winnings tonight. Congratulations, Noki. I just saw, I was like, ooh, Noki made bank tonight. So chat, I want to thank all of our raters tonight. We were rated by tonight Pookie Bear 28, Spleen Be Gone, Real Fun Studios, Professor Astro Canuck, Astronomy Show, and N Simplex Panchico. Thank y'all very much for the raids and for sharing your communities with us. Without y'all, we wouldn't be where we are today. And you know, a raid is an endorsement of a stream. And thank y'all for endorsing us to your communities. Really, really appreciate y'all. It's a huge thing. Tomorrow, we will not be live, but we will be having a Discord watch party on our Discord. So join the Discord if you haven't already. It'll be at 4:30 p.m. Eastern time. We're going to be watching the movie The Meg about the Megalodon shark. We chat wanted a Discord movie night. We got a Discord movie night. This is what won. And so, coolly, we're watching The Meg, starring Jason Statham about a prehistoric shark. Next Thursday, we will chat about the science behind the movie. And by science, you know, obviously mean like de-extinction and that component. Do you have to have... No, so cool. You just have to join the Discord, and there is a movie watch party channel, and you click on the channel, and I'm the one playing it, and you will get to watch it. You don't have to pay anything. All you have to do is be in the Discord. And that'll be at 4:30 p.m. tomorrow. We'll make a Discord announcement when we go live as well. Prehistoric, yes, Shark. It will be about the Megalodon. It's a very goofy movie. I love Jason Statham mechanism. I think he's hilarious. I think he's a great action actor. But that's what we voted on. It's a silly, dumb movie. We're just going to have a good time and enjoy it. And then we'll talk about de-extinction for the science behind the movie next Thursday. Don't Discord with this name. Oh, no worries, Cooley. Cooley, you can join in the channel, and if, you know, we're like, oh, who's this? You can be like, oh, I'm Cooley. And then be like, okay, Gucci. And you know, we usually we have our cameras off and our microphones off, and we just sit back and watch the movie, and we use the chat function, and we usually giggle about the movie with silly lines, and it's just usually a nice, fun time. We're doing it tomorrow afternoon because Lita is coming back. She's doing it overnight with her mom, and she's coming back late tomorrow night. And so little one and I will throw up the movie. She and I will be hanging out, and we'll be watching the movie regardless of at least the two of us. Little baby Alone and I will be watching it. Then we'll be back again on Sunday for Science Art Sunday. Remember, our theme for the month of September is video games. So we'll be doing a video game themed Antonia. We did the God of War Antonia last week, and we started on Resident Evil 4 Antonia of Leon S. Kennedy. We'll finish up Leon S. Kennedy, and then if you get in early for Science Art Sunday, you can vote for what the next video game character is that we're going to work on. I know we have the Halo Antonia ready to go. We've got another one too that's like sketched out and ready. I think I'll sketch out a Kingdom Hearts Antonia. Oh, Final Fantasy Antonia too. So I'll have a couple of options for that. And then next week's schedule is almost identical to this week, except we won't have anything on Saturday. We'll be off on Saturday. But Microscopy is on Monday, looking at tiny worlds under a microscope. Tuesday, we'll be continuing our first playthrough of Pikmin 4. We'll have Wacom Wednesday. We'll be doing art. We'll actually be doing some stuff for TwitchCon on Wednesday. We'll be doing a deep dive on Thursday about the science behind the Meg. Friday is Science News, and Sunday is Science Art Sunday. Dame Karen, you asked about Zelda Antonia. Luckily, Dame Karen. Actually, Dame Karen, I was thinking about that we're going to do... Nintendo will be its own theme. So I want 1 month to be a Nintendo-themed video game. So this month of video games is non-Nintendo. I feel like the Nintendo properties are so good that they should be its own themed month. So that's what we're gonna, and we'll do that at a later point. October is gonna be spooky Antonias. November is gonna be Harry Potter.
Betty a Shark is not tethered by earthly limitations.
What does that mean?
Come.
November will be Harry Potter. December, I think, is gonna be Disney. It's gonna be Disney for December. Tactics, thank you for being here. Guys, I want to do one more quick shout out for one of our friendos, Irish John Games. Let's go follow the lovely Jern, video game developer extraordinaire. Let's go follow Jern. Lion King, Antonia. Wait, Lich King? I do not know the character Noki. But Noki, remember, there is that channel point redemption where you can ask for a custom Antonia, and that doesn't have to fit into any month. So if you wanted a video game one, we would just do that on a Wednesday. Usually is what we'd end up doing. But I'll need to look at that because I do not know what that is. But we'll look into it. Guys, let's go ahead and raid on out for the evening. We are going to raid on over to a gaming friendo. She is playing some... She is very proficient. Tatum! Leave your face, Tatum. She does a lot of retro games. She's currently playing on PS1 and PS2. She's an absolute blast to hang out with. So we're going to go over to Emmy Divergent tonight, formerly Tragic on Twitch. Here are the raid calls. There's that one. You can copy pasta and let her know who sent you on over and tell her about the science we talked about. And if you're following but not subscribed, then have no fear. There's a second raid message for you right on there. Again, thank you to all of our raiders tonight. Simplex, Astronomy Show, Astro Canuck, Real Fun Studios, Spleen Begone, Pookie Bear. Thank you all very much for the raids and for sharing your communities with us. Thank you to the amazing mod team for everything they do. Smikes24, Grimly, NMD96, Andrew CZ, BitGaming, and the lovely Music Girl, without whom none of this would be possible. Thank you all for the viewers for being here and for helping us on this ambassador push, trying to bring more and more science to Twitch. Without y'all, none of this would be possible. Make sure to join the Discord. Movie night tomorrow, or movie afternoon of the Meg. We'll be hanging out in the channels and having a good time there. Make sure to follow us on all the other socials as well. YouTube, Facebook, Twitter, Instagram, Threads. We're almost at 1,000 followers on TikTok, which was our current goal. It's at 50 away-ish, almost 50. So go follow us on there. And I think that's maybe it. Oh yeah, new YouTube video coming out tomorrow as well. So like and subscribe to YouTube. I will see y'all tomorrow for the movie night, and then Sunday for Science Art Sunday. Have a great evening, time zone, wherever y'all may be. Good night, chat.
It's trying.